die, but we'd like to live, and we will always poke fun at physicians, but we'll always pay them when we need them. William Osler, who was one of the great founders of Johns Hopkins, said something that we all know. Many of us on our have spent many years knowing, understanding this. Medicine is the science of uncertainty and an art of probability. And Gordon Clegg, he said, and I think this is ever so true, scientists exhaust themselves, they frequently exhaust their colleagues, they always exhaust their money, but they never exhaust their subject. And the point is that early stage startups really do need to get a handle on this this issue. Money against time and doing the right thing at the right time. Our discussion today will be focused on the toolbox for success in early stage pharma initiatives. We wish to present tools for the early stage pharma operation to use in their journey on the way to the sunny uplands of big pharma. We will have talks from in a very couple of minutes from Dr. Gaby Longsworth, Louis Thomsen, Dr. Leonid Lurya, Itschak Lamensdorf, Idan Dalyot, Moshe Neumann, Zaki Nudelman, Eyal Bressler, and coming in at the end, myself. But before we actually begin in earnest, let's remind ourselves, what is a pharma startup? What is a startup? A pharma or a pharma medical device startup is actually an entity in the very first stages of operations. It's founded by one or more entrepreneurs who want to develop a drug. So a true pharma startup is dedicated to developing a new drug. But what is a drug actually? A drug is an active ingredient in a physical format with a defined method of administration and a medical indication. That's actually what a drug is. And that when you have a drug, then a physician can implement the 6 principles of drug administration. A physician can prescribe the right drug in the right dose at the right time via the correct route and technique to the right patient with the right documentation. That's the whole kit and caboodle of a drug. If you can't get it there, you don't have a drug. So, but we're talking now about very early stage pharma startups. Early stage pharma startups are very different from the vast majority of drugs that are supplied in the world. These are supplied by big pharma. A very small pharma startup is nothing like a big pharma company, and therein lies their weakness and their opportunity. A pharma startup has, all they have is knowledge and a very small amount of time in which to implement that knowledge. The knowledge, they must have a vast amount of knowledge in any of one of the 4 components that make up the drug, either active ingredient knowledge or physical format knowledge, or the method of administration knowledge, or the medical indication knowledge, you know, which culminates in the approved labeling. But at time 0, a pharma startup is like a mouse in comparison to big pharma. But if the knowledge concentration is very high of the pharma startup, this acts as an effector on big pharma. Big pharma will be stimulated to buy, invest, license, or partner. But in order for this to happen, K must be hugely, K must be gigantic. The knowledge in a very short time that the pharma startup has to live, as it were, as an independent entity, must be huge. The knowledge concentration in a chosen area must be vastly greater than that in big pharma, or the a pharma startup will just perish. So the startup has a lot of knowledge and very little time. So what do you need to what do we do with this? You have to learn, you have to manage it. Management is the management of the knowledge and the time, and therefore management translates into team and leadership. And these are not just empty words. The management has to ensure that the knowledge content is high at all times. There has to be a total understanding of what knowledge is essential to be produced within the startup, and what knowledge can and should in fact be outsourced. And a lot of the outsourcing tools are going to be presented. In all cases, it's essential that the company team includes a clinical physician who's an expert in the precise field of the target disease. So, a couple of examples. Let's say a PhD biochemist discovers that a peptide has an in vitro anticoagulant activity. The biochemist decides to form a startup for the purpose of providing a drug to break up the vascular blood clots in COVID-19 patients. I think some of us have heard of COVID-19 by now. Okay, so what should be in the team, in-house, in the garage of the pharma startup? A CEO. There's got to be somebody there who can manage the budget, bring the finance, and manage the IP. The inventor may have those skills or may not have those skills. But if you're interested in anticoagulation in the context of crises in COVID-19 patients, you've got to have in your house, in your tent, an MD hematologist with experience in acute medicine, because the hematologist will set and evaluate the final product goal and characteristics. You've got to have a peptide chemist or biochemist to purify the peptide. You must have a formulation specialist to make the initial formulation. Later on, we'll hear about formulations from Dr. Leonid Lurya. In fact, many of the ideas that I'm presenting here are the results of our discussions. Drug that you need to have in your team, a drug development process pharmacologist or pharmacist, because this is the person who will select and liaise with and supervise the outsourced services, the IP and legal services, the regulatory affairs services, the analytic CROs, the formulation CROs, the preclinical CROs, and the clinical trials. So you need to have someone who can have those conversations. Let's say you've got another kind of startup. A clinical neurologist theorizes that a drug that's known, that's prescribed for epilepsy, might have in low doses a beneficial effect on schizophrenic patients. I think we're going to hear something about second use quite soon. Again, you'll have a similar format for the startup, but you must have inside, in your house, as it were, or in the house, as they say, MD psychiatrists specializing in schizophrenia, because if you don't, who's going to set, evaluate the final product goal and characteristics? Again, you need someone who's going to talk to the IP and legal, regulatory, analytical CROs, formulation, preclinical and clinical trials. So without further ado, dear colleagues from all over the world, in the spirit of Hippocrates, who said, wherever the art of medicine is loved, there is also a love of humanity. And we do this for humanity. There are easier ways to make a living. Let's begin. Let's begin. And I would like to introduce our wonderful and incredibly experienced guest from virtual guest from the United States, Dr. Gaby L. Longsworth, who's the director of Sterne Kessler law firm of Washington, D.C., although I understand she's currently in Florida. It's a great honor, really a great honor for us for me to introduce her. And she will talk now about the patentability of repurposing, new use, and second use. So I'm going to hand over now to Gaby.
Thanks so much, Joe. It's such a pleasure to be invited to speak. I thank my friends at the Bressler firm. Sorry I didn't see you last year. I usually go to Israel at least once a year for the last decade or so. I have a number of clients in Israel, one of my favorite places to go. So hopefully this year. Okay, let me share my screen. Can everyone see that? Can you give me a thumbs up?
Yeah, we can.
Yeah.
Okay, terrific. So today I'm going to talk about strategies and IP considerations for drug repurposing. And let me just move to the next slide. This is sort of the outline of today's discussion. This is one of my absolute favorite subjects to talk about. I could spend hours talking about this, but I have 30 minutes. So I'm going to have to condense it quite a bit, and I hope to answer any questions that we're able to answer at the end. But if we don't, feel free to email me or contact me, and I'm happy to talk some more offline about any questions you may have. So I'll go over very briefly what is drug repurposing for those of you who do not know what we're really talking about, a little bit about regulatory considerations that sort of overlay all of this. Again, for those of you who are not familiar with the FDA's Orange Book, just a very brief tutorial. And then I think the really important part of the discussion, which is the mini case study. You know, what are we exactly talking about? What do patents look like when you have a new use or a new formulation even? I also have a couple of new formulation examples. And then what are the IP considerations that come into play? So we're talking about pharma startups, as Dr. Wyse mentioned. So startups obviously do not have the funds to go into de novo drug development. As we know, de novo drug development is extremely costly. 1.3 billion is sort of an average number. It's ridiculously high, and it's very time consuming. It can take, on average, 16 years to develop a brand new drug. Which is why developing either a new indication or a new formulation of a known drug is very attractive, because this can be accomplished piggybacking off of the initial studies, can take anywhere from 2 to 5 years, and is significantly less expensive. So when we talk about repurposing, we're talking about either reformulating or repositioning. Reformulating obviously means what it says. It's just a new formulation, a different formulation for the same drug. Whereas repositioning, when we say repositioning, we're typically talking about a new therapeutic use for a known drug. So you could target either new patient population, as I said, new indication, new dosage form, maybe new route of administration, maybe different dosing regimen. All of these would be falling under repurposing. So typically in pharma, when you are trying to get a new drug on the market, there are broadly three different ways to accomplish this. The new drug application, which falls under section 505(b)(1) of the statute. As I said, very expensive, very time consuming, contains full reports of safety, efficacy studies conducted by the applicant. The application that we're mostly focusing on today is what's called the 505(b)(2) application, which again is part of the statute, and often is referred to as a paper NDA. So it's a modification to an approved drug, and you typically rely upon one or more studies that were not conducted by the applicant, but that were conducted by the NDA holder. If there is additional data required by the FDA, then you will have to do additional studies, but sometimes this is very limited. It just depends on what the actual 505(b)(2) application is to. And this can actually be branded as you can brand it as a drug. You can list it in the Orange Book. You can get your own patents. You can list the patents in the Orange Book for a paper NDA. And then the last one, the ANDA or ANDA, is an abbreviated new drug application. I'm sure you're all very familiar with the process of sort of getting generic drugs on the market that proceeds through the ANDA. I do a lot of work for Teva on both the branded side of things and also on the generic side of things. So very well versed, Israel very well versed in both sides of the equation. And for ANDAs, you're basically making a duplicate of the original drug. You're relying on all the studies. It has to be identical in every aspect in terms of active ingredient, the route of administration, the strength, the dosage form, the use. We're not going to be talking about ANDAs today. We're really going to be focusing on the paper NDAs. But just wanted to highlight the different types of drug applications and make sure that we all are on the same page. And some of the slides, I'm going to skip over a little bit faster because I assume that you will all be able to get a copy of the presentation. So the only thing I wanted to mention here for the 505(b)(2) is that the applicant relies on safety and effectiveness data of what's called the reference listed drug. And the reference listed drug can be a number of things, as you'll see in one of the examples. It could even be a discontinued product that is your reference listed drug. Okay. Okay, so we talked about ANDAs. I'm just listing it for information because we're not focusing on that. Yeah. So now exclusivities. Why do people even want to get new drugs on the market? Well, first of all, you want to get new drugs on the market, obviously, to help patients, but also you want to get some sort of exclusivity period, right? To recoup all the investment that you put in. Besides IP, besides patent, you want to have some sort of exclusivity. So these are the different types of exclusivities that you can receive in the US from the FDA. I've listed them here. There's orphan drug exclusivity, which is 7 years, which is very valuable, but it's pretty limited, right? Because it's only given for conditions where there's less than 200,000 patients. New chemical entity is a super valuable one. You get 5 years. Basically, what that means is for 5 years, the FDA cannot approve a generic competitor's ANDA unless they file a specific, if they go through a specific pathway where they can start at year 4. But you can imagine how much you can recoup of your investment in the first 5 years. There's other types of exclusivity, like a new use, new dosage form, which are 3 years, and then you can also get pediatric exclusivity of 6 months added. The Hatch-Waxman exclusivity that I mentioned here is just for ANDAs, and in that case, you can get 6 months of generic exclusivity. This is just showing a quick comparison, visual form of what I just said. And then again, the advantages that we talked about for 505(b)(2s, especially for pharma startups, where is a good place to start? A 505(b)(2) might be a good place to start because you can piggyback off a lot of the studies, and depending what it is, if it's the simple dosage form like an oral tablet, a lot less is required than if it's an IV, for example. But it's a way to get some sort of exclusivity, get your own IP, and come up with something that could be truly very valuable in generating a lot of revenue. As opposed to an ANDA where you have a lot of competitors all at the same time. And here's just a few examples of some of the 505(b)(2) strategies that we have seen over the years. So you can see what the reference drug was in this chart, what the 505(b)(2) drug is, and what was changed, whether it's a new dosage form, new formulation, a new combination, and the kind of revenue that was generated. Can be pretty significant. Okay, so quick Orange Book primer, for those of you who don't know. Back in 1984, when the Hatch-Waxman Act was came about in the US, Congress also made required the FDA to make publicly available a list of approved drug products with therapeutic equivalence. And this list was published at the time in an orange book. It was a book with an orange cover. And they have several different books these days. Like for vet drugs, it's a different color. There's the purple book, there's the red book, etc. And the orange book will include any approved NDA, 505(b)(2) or ANDA product. It also includes drug patent information, patent numbers, etc. And it's electronically available. And I'm not even exaggerating when I tell you that I am on the orange book every single day. It's so incredibly valuable. It has tons of information. So very quickly, this is sort of what it looks like. I put this here so you can see the URL. For those of you who don't know it, I have it bookmarked. You can search in many different ways for pretty much any drug. This is an example of paclitaxel. And you can see the very first entry is Abraxas, is a paclitaxel product, Abraxane. It shows you the NDA number, N0216660. It tells you that it's a suspension, that it's an IV infusion. It gives you the strength. And you can see on the right where it says RLD, it's the reference list of drug. You know it's an NDA because it starts with N, the number. And below it is all these generic versions, which all start with A for ANDA. And there's a ton of them. And they're all the same. Well, they all have a different strength. You see 6 mg per vial, as opposed to the reference list of drug. When you search for a drug, this is sort of what you get. It tells you a lot of the information that I just showed you on the other page, but it tells you when it was approved, the active ingredient, etc., dosage form. And then below you can see where it says patent exclusivity information. If you click on that, it shows you all the patents that are listed in the orange book that cover this product. And as any smart innovator will do, you try to get as many patents as possible to block competitors for as much as possible. The more patents you have, the more complicated it is to invalidate and to get in there. If you have a large number of claims, that also adds another layer of complexity. So there are a ton of patents here that shows you the use code. So if you hover over each of these, you will see the use codes, which is what this was approved for. And if it's a drug product, it will say DP. If it doesn't have that designation, then it's a use patent. And then also see all the expiration dates. So tons of information to get you started. If you're interested in a certain drug, well, let's just see how well protected it is. And if you see something like this, you probably don't want to go there unless you have deep pockets. Okay, so now let's go to case studies. So first example is a drug reformulation example. I thought it would be helpful to also show drug reformulation, because sometimes for drug startups, pharma startups, it's actually an easier way to go to a reformulation approach rather than a new use approach. And this is for amantadine. This is actually one I worked on for my client, Osmotica. But what is the story here? So amantadine, old drug, well-known drug, used to treat Parkinson's disease. It was initially marketed by Endo as an immediate release tablet and as a syrup. And it was called Symetrel. So that was an NDA by Endo. Then Osmotica's competitor, Adamas, marketed its extended release oral capsule, Gocofri, which was a 505(b)(2) application because, right, it's an extended release oral capsule. The original one was an immediate release tablet or a syrup. So this is a new formulation. So piggybacking off of the initial studies, clinical studies by the innovator, Adamas received approval for their version by filing a 505(b)(2) application. My client, Osmotica, also made a different version of amantadine. In fact, they made an extended release tablet that had an immediate release coating. They also filed it as a 505(b)(2), but instead of using Simitrel as their reference listed drug, they used the immediate release tablet Simitrel. They didn't use that as their reference listed drug. They used the syrup as the reference listed drug. Here's just a quick summary, sort of putting it all together. Because you can use as your reference listed drug any form that was previously approved, whether it's still on the market or whether it was discontinued. So here's just a quick comparison of everything. And you can see for the Simitrel product was actually withdrawn by Endo at some point. Gocofri, just in the first year, made $54 million in sales.
Wow.
Exactly, pretty significant for sort of a reformulated drug. And then my clients, we don't have the sales data here. It was significantly less than 54 million because it was the second one on the market. But they still made some money off of it and actually made a deal with Adamas recently. And here's just a quick comparison showing you the different formulations. So Simitrel, the oral tablet versus the syrup. Different concentration. Either one could have been used as the reference listed drug. My client used the syrup and Adamas used the tablet as the reference listed drug. So what about the patents? So when you look at the Orange Book for Gocofri, you see that there are at least, this may actually be outdated, there's at least 15 patents covering this new extended release formulation. And they have a number of patents that are to uses, so the use of this new formulation, and I've sort of bolded the limitations that really got them through the patent office. That was different. Basically, they're claiming, you know, a subset of formulations, amantadine formulations for this use, the subset being that it has to have these specific PK parameters. That's always a clever way to get patents in the patent office with PK parameters, because it's difficult for the examiners to show that this was in the prior art. So here's one example of a patent that expires in 2025. Here's another example of a formulation patent that Adamas has, which is to a dosage form, and you could sort of see the language there. Okay, and I'll talk about sort of the IP perspectives, like how do you get something like this through the patent office when there's so much known already about the drug. Here's an example of one of the patents that we got for Osmotica. Osmotica already had 2 patents to the actual formulation, very, very narrow patents. We got them using Truck 1 approach, which is an accelerated examination in the US. We got them 7 new patents fairly quickly. This is sort of what these patents look like. You know, covering the extended release and the immediate release components, and also having a PK parameter in there. So both 505(b)(2) applicants had numerous patents listed in the Orange Book, which then competitors would have to attack. So that's the first example. Here's the second example, also a reformulation example. The third example will be a new use example. So this is another one. This is Wyse, and Wyse did both these are actually NDAs. They filed 2 different NDAs for, you know, strategic reasons, but they could have easily filed a second drug product as a 505(b)(2). Well, the initial product that was on the market was the Effexor, which are immediate release tablets of venlafaxine, and this is for depression and among other things. And you can see the sales data. This is, of course, pretty old data, but it was over $2 billion in 2001. Wyse started realizing that, you know, some of the patents were starting to get near their expiration dates. So they came up with this clever strategy to make an extended release version of venlafaxine, and they made extended release capsules. Sort of overlapping strength. So like I said, they filed it as an NDA, but they could have filed it as a 505(b)(2) because it's a new formulation, new strength of an old drug. And they got approval a few years later, And they actually made more money with the new strength, new formulated version of venlafaxine than they did with the original one. What are some of the patents that they got? Well, first, the original compound patent expired in 2008. And by coming up with this extended release version and getting new patents, they actually were able to extend their monopoly by about 9 years until 2017. So it's a very it's a very common strategy, and it's a good strategy. And you can get patents out of the patent office. Now, when you're doing an ER, extended release formulation of a known drug, of course, there's going to be, it's not going to be easy getting it through the patent office. It's possible, but you will have to narrow the claims in certain ways. And you could see the example here of the very first patent, the extended release formulation of venlafaxine. It's very narrow, right? You have particular percentages of the excipients. You can design around such a claim if there's some wiggle room. But they did get new patents. They can list them in the Orange Book, and they can still block competitors at least for several years. A method of treatment patent, which is the second example, is not as narrow, and those actually, to me, provide more protection in some ways than the formulation. As long as the claim is exactly follows exactly the indicated use, it's very difficult for a competitor to get around a method of treatment claim. And same for the third example. Okay, the last example is a drug repositioning example. So this is for a new use. And this is one of my favorite examples because it's just so clever, and it was just made on an observation. So it's about finasteride, which originally was marketed as Merck's, it's a Merck product, as Proscar, which was for benign prostatic hyperplasia in men with an enlarged prostate, and it was 5 mg tablet. Just a simple 5 mg immediate release tablet. This was approved in '92 and made a lot of money right away, still making a lot of money, as BPH is obviously very, very common. But they noticed there were some observations made that men who were taking this drug, Proscar, for BPH, started growing their hair back. And Merck, obviously being a very, very experienced company, realized that they could lower the dose from 5 mg to 1 mg, same tablet, and market it as Propecia for male pattern hair loss. And they made even more money for hair loss. They're still making a whole lot of money for hair loss. So this is a very, very nice example, I think, of really being on top of other potential uses that may come out of the initial use. Same formulation, just reposition it. What do some of the patents look like? So here, they're all method of treatment patents that I'm showing because, again, having the formulation being so simple, it's very difficult to get new patents on an old drug with a simple formulation, but the methods are very valuable. So for Proscar, it was the method of reducing the risk. There's different ways, obviously, to claim it. You can claim a method of treating BPH, but they also had different claims to reducing the risk of acute urinary retention. And then some of the examples of the use claims for Propecia, method of treating male pattern baldness or treating androgenic alopecia. So valuable patents were obtained that were blocking and had a lot of value. Okay, so in the last few minutes, let's talk about the IP considerations. And I should say, for some of these formulations, like the extended release formulation, that's probably fairly simple, but you can imagine that if you are trying to reformulate, that there's going to be some formulation challenges. And as Dr. Wyse said earlier, you've got to have the right experts. And I know Dr. Lurya will talk about formulation challenges in drug development, but this would inform you as to which way to proceed. If something is extremely difficult to formulate, that could be good because you're blocking a lot of competitors or others who may want to follow because it's so difficult to formulate. But on the other hand, you may run into a lot of issues as well. So it's a very, it's a balanced approach. As you saw from some of the examples, you can obtain patent protection for new formulations for new uses. Of course, it depends on what was already known and whether, there were already suggestions or the mechanism of action was known and it was pretty clear, sort of extrapolating from that what the other uses might be. So just as a starting point, if you are interested in this area, I think it's fairly important to do both a prior art search and a freedom to operate search, just to figure out where the holes may be and where the opportunities may lie. I know it can be more expensive to do it this way, but in the long run, you will actually save yourself a lot of wasted time and money, sort of going around in circles, not knowing exactly what the roadmap is. So once a drug is known, obviously you cannot get a composition of matter claim again, unless it's perhaps a new polymorph or some other new form of the drug. But formulations are typically, novel formulations are typically easy to get. Well, let me not say easy to get. You can file them, but they can be easy and they can be difficult. It depends. The more art there is, the narrower the claims will be. The narrower the claims can have to be, the easier it is to design around. So there's some drawbacks there. Getting patents on a new use can be much easier than getting a new formulation, with the caveats that not much is known about the mechanism of action, etc. And of course, if it's a very different use, that makes things easier. If it was first approved for acne and now you've figured out that it can help with depression, those are fairly extremely different. But if it was acne and now psoriasis, we're sort of a little closer, kind of more in the same space. So again, it depends on what the new use may be. But typically, if it appears that it would have been obvious to go, say, from acne to psoriasis as the new use, as long as you have some new unexpected result or something surprising that would not have been foreseeable, you could present that to the patent office, and typically that would get you sort of around the obviousness. And here's just some unexpected results that may help. If it works surprisingly well, or if it's at a surprisingly low dose, if it's a combination, is there synergy? Is there a different target? You get the point, right? When you're doing this research and you're in this space, these are sort of the things that you want to look at and see, anticipate sort of the rejections that may come along. And if you have an experienced patent attorney, they will be able to guide you, point you in the direction of what are the things to research, what is the data that you might want to include in the patent application if you have it, where are the angles to pursue. Okay, I think that's the end of my presentation. I think I'm way out of time. Happy to answer any questions, and I'll stop sharing.
Gaby, thank you very much for that interesting talk. Now we have a couple of questions, but unfortunately we don't have a lot of time, so let's focus on one question. Have you encountered with natural products labeling for specific health situations? Any idea for the best strategy?
Sorry, can you repeat that question?
Yeah, of course, and you can see it in the chat as well. Have you encountered natural products labeling for specific health situations, and any idea for the best strategy?
So yeah, natural products can be tricky because of course, the patent eligibility requirements in the US that you can't get a patent to a product that's found in nature. But there are ways to get around it if it's in a formulation, depending on what the formulation is. Obviously not the natural product plus water, but something a little more more than a simple combination. You still will be able to get a natural product patented in a formulation, and of course having a new use with the same angle will get you around that as well. There's a whole lot more to say about the subject, and I'm happy to answer a little bit further whoever asked that question if you email me.
Excellent, thank you very much, because we have some other questions, additional questions, but unfortunately we do not have the time. So with your permission, we will forward you later on the questions from the audience.
Thank you.
Okay, excellent, thank you. Now we are moving on, and we have the great pleasure of welcoming Ms. Louise Vibjerg Thomsen, a commercial counselor and senior innovation officer from the Danish embassy in Tel Aviv and from the Innovation Center of Denmark in Israel, who will speak about the Danish pharma startup and academic ecosystem. Louise, please.
Yes, thank you very much. I'm just going to see if I can get the screen to work. Do you see the screen? Great.
Yeah, we can see.
Yes, so thank you very much for inviting me just to give you a short intro to the Danish pharma ecosystem, just for you to see if there are any particular types of collaborations that might be relevant for you in your developments. I don't know how much you know about Denmark or Danish pharma, but at least in Israel, I know a lot of Israelis know the bridge from TV. So that at least is a good symbol also for talking about Danish pharma. Which is very much focused on collaboration of the ecosystem. And this is extremely important if you would like to engage with some of the Danish stakeholders. But before I dig into that, I just wanted to shortly introduce myself. I'm the Senior Innovation Officer at the Innovation Center of Denmark in Tel Aviv. Denmark has 8 innovation centers around the world, located at the Danish embassies. And these are in places that we see as innovation hotspots of particularly relevance for collaboration with Danish stakeholders of all sorts, whether it's investors, startups, research, or other types. So we also have centers in Boston, in Shanghai, in Seoul, and other locations. Now in Tel Aviv, one of our main focus areas is the area of life science. And that, of course, is because it's one of the main sectors in Denmark, as well as in Israel. And Denmark is very strong in life science, both in terms of research and in terms of industry. And in particular, Denmark is considered a powerhouse in pharma. And as you can see, we rank top in a number of issues such as global biotech. We're second in the world. We're first in clinical trials in per capita in Europe and in research and so on. This area continues also to grow in Denmark. Actually, pharma has tenfolded in the last 10 years in terms of turnaround in Denmark. And particularly interesting are, of course, the issues of research and clinical trials. If we look at the beacons of Danish pharma research, they are particularly in cancer, diabetes, CNS enzymes, infectious diseases, and heart diseases. These, of course, reflect a little bit also what some of our leading corporates are looking at, but also what our researchers are particularly strong in, both in universities and in the hospitals. In addition, one of the new areas that Denmark is very much looking at is the issue of medical cannabis, if that is what any of you are also looking at. And Denmark is currently working to become Europe's leading ecosystem also for medical cannabis. Denmark made it legal to produce medical cannabis about 1.5 years ago, but also to subscribe medical cannabis, so both in terms of industry and the healthcare system. That is very much expanding. In addition, I think it's important to know when you're working with the Danish ecosystem, as in many other parts of the world, there's an increasing focus on the green agenda. And also for the Danish corporates, when they look for collaboration with startups from around the world, particularly also in pharma, the issue of have you considered the green aspect in your development and production thoughts will be an important factor in attracting the attention of Danish pharma and Danish investors as well. Because it is an issue that's growing very much in Europe, and Denmark is determined to very much run with this agenda, and is also very determined that pharma will take this as a top priority in the near future. The other issue is the issue of clinical trials. Denmark has very fast, safe, and cost-effective drug development. And it's one of the fastest countries in approving clinical trials, which is one reason why it is very attractive to conduct clinical trials in Denmark for startups. Another reason is that Denmark has a national healthcare system that's a little similar to the Israeli healthcare system. And we have a public EMR completely digitalized that follows each citizen from embryo to grave. So it's a complete data set. And we have a data set that is goes back a lot more years even than Israel. So we probably have one of the best data sets in the world. And we also have 500 different biobanks. So in terms of data, Denmark could also be an interesting place to explore clinical trials. And because of that, Denmark, about 1 year ago, established a unit called Trial Nation Denmark. And it's a unit that provides a entry point into conducting clinical trials in Denmark, which means that this unit will help you identifying Danish investigators, getting national response to request, estimating the patient eligibility for your trial, even helping you find relevant partnerships with hospitals and scientists. And they do all of this free of charge. So if anybody is looking towards Denmark, I think it's a very good entry point to not use too much time yourself trying to figure out the system. Now, many of you probably know the large Danish pharma companies, Novo Nordisk, Lundbeck, Leo, and so on. And they are, of course, very important to startup companies. But in addition, Denmark has a very vibrant academic and startup ecosystem. And in addition, Denmark has a lot of middle-sized companies that probably, at least in Israeli scale, might even be considered big companies. And they should not be disregarded if you're looking for partners in Denmark. Because they do a lot of very extensive research, and they're very much interested in international collaboration, and they have the resources to lift international collaborations. So it's always good when you're looking at partners in Denmark also to consider the middle-sized companies. In addition, many of the Danish corporates, they are actually run as foundations. which means that the idea of doing that was that Denmark didn't want companies to leave Denmark. They wanted companies to stay there and do their development in Denmark in order for us to have a strong life science ecosystem. And so these companies are owned by foundations that provide a lot of financing for doing research and development in Denmark. And they are very focused also on having some kind of social accountability as well, and providing back to society. And these very large foundations, they are also open to collaboration with international startups. So they do provide research grant and other grants to international projects that they think could have a good social impact. So they would also be worth considering. In addition, we have our hospitals that do a lot of very high quality research along with our universities like the Danish Technical University, Copenhagen University, and so on. And in Denmark, about 50% of the Danish pharma startups are actually spin-offs from hospitals and academia. And if we have to be honest, then the Danish hospitals and corporates until now have been quite conservative and haven't engaged a great deal with industry and external innovation. But that has very much changed, and I'll touch upon that a little later also how. But both corporates and hospitals today have established infrastructures for collaborating with startups and researchers, also internationally, and they run tailored programs that any international stakeholder can tap into. I think when talking about Danish startups, I think it's important just to point out that the Danish entrepreneurial dream is a little different than in many other countries, because most entrepreneurs, also in pharma, their dream is not so much to make an exit. Of course, there are some that would like to make an exit and go on and be serial entrepreneurs and so on, but most Danish entrepreneurs, their big dream is to be the next Novo Nordisk or Pfizer or Merck, because they want to have a great impact on society and they want to be able to continue to develop new solutions within a good framework. And so also the whole Danish ecosystem is kind of built on this idea that you want to help companies also think of whether it's worth it to build bigger rather than just exiting, so that we can have more of a vibrant ecosystem also within pharma. But this also means that in Denmark, there's a great focus on collaboration between the industry, the hospitals, and the universities. And so all of these have actually joined together because in Denmark, we like clusters. We think that they're a great way to collaborate. And we've formed in Denmark, we have the Medicon Valley, which includes also the southern part of Sweden. So in an area that's smaller than Gush Dan in Israel, you have over 12 universities and 35 university hospitals and 600 companies and so on in a very small space. And they are doing a lot of collaborations. They have formed an alliance called the Medicon Valley Alliance, where they sit and they collaborate on a daily basis, where scientists can pose issues to companies, asking them for data or for resources or for ideas, and vice versa, which gives a more vibrant process for drug development. And this is also a great entry point for foreign stakeholders wanting to engage with the Danish life science ecosystem. Also because if you're already connected to one of these parts, then you're connected to all of them. And you can then also be part of receiving data and information from any of the stakeholders that are part of the Medicon Valley Alliance. And they're very much open also to international collaboration. And so the whole idea here is that the whole is greater than the sum of its parts. And it's very much part of what Denmark believes also in terms of research and development. So I just wanted to close up with saying, if anybody that's really looking towards Denmark for collaborations, there are a couple of things, places that could be a good entry point. There are multiple partners that could be approached, such as the Medicon Valley Alliance. On the 1st of January, Denmark also established the Danish Life Science Cluster, which really brings all of the clusters in Denmark within all areas of life science into one cluster that covers all of Denmark. You could also explore as a startup some of the incubators of the corporate companies. The most well-known, which I think is really could be interesting, is the BII, which is the Bio Innovation Institute, which is the incubator of Novo Nordisk Foundation. They don't just look on diabetes, which is what Novo is working with. They look across all therapeutic areas, and they run a program for startups of all stages for 3 years, where you can be located in Copenhagen right next to all of the big pharma companies. And they help you through some of the development stages, and also exposing you to some of the Nordic investors. And they're very much open to receiving international startups at the moment. And then, of course, if you're doing clinical trials, to get in touch with Trial Nation Healthcare Denmark. And there are a lot of startup programs in Denmark for pharma companies. Then I think I would also just like to mention that hospitals in Denmark, like I said before, are now creating innovation units. And in Denmark, we just had a reform where we decided to close a lot of our hospitals and to build what we call, in a not very good term, and that will change, but at the moment they're called super hospitals. So we built 5 large hospitals in Denmark. And the idea is that these hospitals will provide all types of examinations and all types of treatments and surgeries under one roof, so that a patient can get whatever kind of treatment they want in the hospital, so you don't have to move them around. But this also makes it very interesting for industry and for researchers, because it opens for the interdisciplinary research and development that is becoming increasingly important. And these hospitals have now just opened innovation units where they run startup pilot projects, and startups can collaborate with them. And they have a very simple form that you fill out and send, and then they find the relevant investigator or physician to collaborate with. And of course, we have the Horizon Europe funding. We hope also Israel will join Horizon Europe. There's a great focus in Horizon Europe this time also on pharma development, particularly within oncology, will be one of the main areas. And then Denmark is a great European entry into the entire European area. It can be difficult to collaborate directly sometimes with Germany and France and some of the others because of the regulations and the frameworks, but it can be done through Denmark that has done a lot to provide an international framework in terms of approvals, permissions, and taxation. And then maybe just to round off, of course, the Innovation Center in Tel Aviv is here also to help anyone to connect to the Danish life science ecosystem. And we're running this year quite a number of programs that anyone is invited to join, such as startup testings in Danish hospitals. We're looking on doing a bioconvergence program with focus on pharma, which will include different kinds of research grants and scholarships. We'll also be forming a hospital alliance for exchange of startups and researchers between hospitals, and we're doing a Danish investor tour for Israeli startups looking for Danish investments. So I think that was just a very short intro to Danish pharma, and you're always very welcome to reach out to me if you're interested in identifying potential partners.
Thank you very much, Louise, for a very interesting and stimulating discussion and presentation. And since we're a little bit short of time, everybody who wants to reach Louise, the email is here. And if not, you can email us and we'll email Louise email. And I'm really, really glad to present Dr. Leonid Lurya, the Chief Technology Officer of Chilion, Israel. And his talk will be on formulation challenges in drug development. And Leonid, the virtual floor is yours. Leonid?
Thank you. Good evening, ladies and gentlemen. And first of all, I would like to thank organizer for inviting me to such meetings. Now, sorry. Okay. Here we are. Oops. Right. Can you see my presentation?
Yes.
Okay, so the topic of this presentation is formulation challenge in drug development. And in this presentation, I would like to highlight not specific method to solve formulation problems, but mainly set out the basic principles that an inventor should to know and use for most successful, let's say once again, users, specialists in formulation development to resolve these problems. Let's start to look first for the, sorry, for the drug development process in general. Actually, it starts, the first three stages are most important for us, which lead to the approval. And the process begins when using a biological source or chemical synthesis technology in development process, we find a series of potential active compounds and then select through the screening process the lead compound. Then, as you can see from this slide, in the next preclinical development stage, these lead compounds should to overcome for their furthest studies and development, like proof of mechanism of action, animal pharmacokinetic pharmacodynamic study, animal administration, administration, distribution, metabolism, and excretion. We have to evaluate the safety of these potential compounds, found the proper dose ranging, drug delivery method, definitely prove at the end some efficacy, and to develop all clinical validation method that will allow us to come to the next and final for us, clinical development process stage, which includes a very known phase 1 to 3 study in which we have to demonstrate now on the human being efficacy, safety, pharmacokinetic study, and find the final proper dose and bioavailability parameters to get to receive approval of the new drug. Let me to describe this process the moment in a view of formulator, and based on my experience working in pharma industry and pharma startups for more than 20 years. Let's make some kind of example. Let's look at some kind of example when you, the startup decide to develop the anti-cancer drug. The development based on the well-established scientific basis of malignancy mechanism, even on molecular level, and we can even consider what should be type of molecule that have to work as an anti-cancer material. And we even proved this in a screening test and found the lead compound efficacy of this compound is about 80%. Then we continue to work on the next model, like cancer tissue, and we can inject this material inside this tissue and sees very well as an effect. It might be slightly low, but we can see it. But when we start to go to the more complicated animal model, when we not know exactly where is the cancer cell localization, and we start to see degrees in efficacy of this material, and if finally in the clinical stage of development we will reach the let's say 20–30% of efficacy, it will be proof as a drug. So now we raise the question why we have such change through the drug development process, decrease in the efficiency. And it doesn't mean that the molecule is not work. We can prove it in the cell model. The answer is clear. The drug is not appear in the proper, in the right time, at the right place. And the main problem is that the delivery system, delivery method is felt in this situation, or maybe it's natural that it can't be more efficient if we will take in consideration the complexity of the human being and restriction of toxicity of this active molecule. Thus, to my point of view, it should be demonstrate the importance of formulation in drug development process. Now, let's consider the different types of innovation approach for new so-called new pharma development for a startup. It might be three types. One innovation is new drug discovery. It means screening and development of a new molecule entity or new therapeutic biological product. The other type is new medical device or new drug delivery system. That may be more specifically described in the following slide, like a medical device is any device intended to be used for medical purposes. Medical device benefit patients by helping healthcare professional provide diagnosis and treat patient and helping patient overcome sickness or disease, improving their quality of life. So it's like here we have a general definition of medical device, and we are talking about drug delivery system. So we can say that drug delivery refers to approaches, formulation, technology, and system for transporting a pharmaceutical compound in the body is needed to safely achieve its desired therapeutic effects. Drug delivery is approached via a drug chemical formulation, but in many cases it also might involve medical devices or drug device combination product. If we will consider the role of formulation in all these type of innovation, we can clearly see that actually in all of these type of innovation, formulation is a very important tool for development a final product. In addition, when we have a deal with a drug delivery system, this formulation may be as not only maybe not only an instrument, it might be also subject of innovation. It's a formulation technology which actually provide this new efficient product. Now, what formulation challenge we meet in drug development, if we will consider for our purpose of our discussion that formulation is an instrument. Actually, here I would like to clearly define 4 important challenges. First is we need proper specification. Next, second, we need to keep it constant. Formulation development should be constant through all the development processes. Also, it's very important that through all development, a formulation expert is in full interaction with all development team. And also it's very important to keep in mind that at the end of the formulation development will be a very important stage of technological transfer to the industrial production site. And let's look all these 4 challenges in more details. First of all, proper specification. According to my experience, it's normally what happens that the formulator or formulation advisor meet with such requirement. Well, you are an expert. Please develop the formulation as you know, as good as possible. And it doesn't work. Formulator need to know, first of all, very clear definition of the dosage form, which the inventor with all his proper team, as Dr. Joe Wyse presented before, all these experts have to very clear define the dosage form with very well defined, at least theoretically, concentration range of active material in formulation or in drug delivery system. It should be very clear defined what is the targeting site of the active material, where he have to go, which part of the body it's probably have to escape, and what also is a specific problem which relate to this to the active material. It has a problem of solubility, bioavailability, what's going on with toxicity and safety requirements. And definitely it's not so important like we say, oh, the dosage, its formulation should be stable, or there is a regulation requirements. This formulator definitely knows. What's important is what we say before, formulation, formulator need to know. These four very important parameter of specification. Now, when we are talking about consistency through the development process, I mean the following. Our aim as a formulator is a successful clinical trials. So from the beginning, we have to keep in mind that if we are talking about efficiency, or safety, it should not be different formulation, because it's easy applied to efficiency, to study efficiency biological model with one application and safety with another, and then we have a very quick and maybe good result, but then we will meet a very big problem when we come for regulation approval. It appears that it's different formulation, a lot of questions of formulator, and sometimes it's the companies start to do this research from the beginning. And if you will not keep in mind these and not work in very clear cooperation with formulator, which know how to reach this integrity, so it will be very important for successful in development process. The next, when we are talking constant interaction with whole development team, it always looks like a ping-pong process. You should to know that, I mean, now I apply to inventors, that when if even you give a very clear specification, it not will be happen at the end that formulator will go to his laboratory to close himself for 2 weeks and then make a focus and will bring you a good formulation. It always might be have the process of improvement, of final tuning, and all these small but very important changes, which is going from here to there and from there to here. Now it's we have new formulation. How do you like it? It's work. Suddenly you find out that you need different to change the requirements, and you have to you should to know this is normal. It's much better to spend more time and efforts through this process than to get some problem at the next stage of testing of formulation. And suddenly it might be a very unusual problem arise in the formulation development, and in this case, in this situation, he also needs to be in contact with the whole team. I can give some personal example, like new startup applied to me for development of dermal formulation of new anti-cancer drug for skin cancer. It seems very easy task, and after 2 weeks of work in laboratory, I present let's put in this way, formulation was ready for next testing, but it's normal. I decided to prove that the concentration in this formulation is more or less according to the requirements. And to my surprise, it appears that there is no active material. Analytical result was 0. And after a couple of weeks, of the very unusual investigation, it appeared that actually the material which was synthesized was 99.2% of purity. There was a little amount of impurity, each was less than 0.5%, and according to analytical requirements, it should not be to estimate. But what it's appeared that actually in purification, the company which make a synthesis also didn't do this test, and 0.03% of the impurity appears to be a catalyst material which remain in this material. At the moment it will met water phase, it immediately start the reverse reaction, and in a couple of hours the amount of the material was 0 in the formulation. Therefore, this example is clear demonstrate that especially in where is a startup with a new and theoretically excited material, we should be very, very careful, and each member of the team which is responsible for their own part in the development has to do his work as better as possible. And as I mentioned, in very close cooperation with all other members of the development process members. And now actually the last and very important part in drug development process, which is main responsibility of formulator expert, is keep in mind that the success of the actually inventor or active materials will be proof in clinical trials, but success of formulator will be proof in technological transfer to industrial site of production. And here is 3 important aspects which formulator should take in consideration from the beginning. First of all, this is a technological aspect. The formulation should be as simple as possible. We can develop in laboratory very nice and very scientific, excited type of delivery system. But then when we will go to and try to produce this formulation, we can get a lot of industrial problem to realize this scientific idea. And the best example in here is the liposome technology. It's there is a lot of patents and scientific paper for different type of a very excited liposome particle. And it might be looks like a Sputnik, but at the end of the day, it's almost impossible to develop the industrial technological process which might be easy produced in laboratory. The next, from the beginning, we have to start to use industrial approved excipients. There is a lot of excipients which we can buy cheap and easy in Sigma, Merck, and have get a good result of solubility or another formulation reach another formulation requirements, but these excipients are not used in industry, and it might be faced the problem for technological transfer. The same story is for industrial acceptable method. For example, it's very easy and efficient to use ultrasound technology in laboratory, but you should to know that there almost not apply for industry. It's very expensive method for manufacturing. The same story, we can easily use the centrifuge to separate material. In industry, the main there is the centrifuge, but it might be very expensive. Okay, the Iranian physics use centrifuge to get a nice formulation for the nuclear weapons, but it's not what we plan to use in pharmacology. It's expensive. The next aspect is
sorry, Leonid, you have 2 minutes left. Sorry.
Yes, but in the last slide I will be
okay,
fine.
Perfect. Sorry.
Thank you, Rita. The next aspect is regulatory aspect. Is as I mentioned, used industrial improvement excipients, and pay attention that there is specific requirements. GPM, ISO for industrial, and it's much better if formulator will be taken in consideration when he starts to develop his formulation in terms of composition and method of preparation. And the last aspect is economical aspect, to make the cost optimization of using excipient and cost optimization of the technological process is also very important, and it's a responsibility of the formulator. And I think that the pharmaceutical startup will provide this product, not only excited from the in terms of efficacy and safety, but also will be suitable and efficient for industrial production. It will be a win situation. Thank you very much.
Thank you, dear Leonid, for very interesting and useful presentation. And now I would like to present Dr. Itschak Lamensdorf. Is the founder of Pharmaseed. Pharmaseed is a preclinical CRO in Israel, and he will talk about preclinical studies, do's and don'ts in pharma R&D. Itschak, the floor is yours.
Now, do you hear me?
Now we can hear you.
Okay, can you upload the presentation that I have, or
do you have it?
I think that Joe has it.
Okay, Joe, in a minute.
I can sing meanwhile.
Sorry, what did you say?
I said that I can sing meanwhile.
You can sing. I think it's the
cultural
best
break.
entertainment we can have in these lockdown days. If you sing, it will be a very good.
Okay. Nice. Okay, so first I would like to thank for inviting me to give this short presentation. During the next, and I will try to be short, during the next 10-15 minutes, I will try to provide some hints as to my understanding as to the role or the integration of the preclinical work within the general development of product, and give some, again, from my humble opinion, what are the do and do not key point. Next slide. So, can you move to the next slide, Ravital? So to start with, the question is why? Why to conduct? What is the rationale of preclinical work? And the why is as a result of few reasons. Why? Because it is a must. Without appropriate preclinical package, you won't be able to move forward. Without appropriate preclinical/discovery package, you won't be able to raise necessary fund. Another why is because we want to predict efficacy and safety. And based on this prediction, to assess what is the therapeutic ratio. At earliest stage, we need the preclinical work to enable us to prioritize, to move between heat type of molecule to the lead one. At a later stage, it's becoming more issue of optimization, the formulation optimization, the duration of administration, the mode of administration, etc. And otherwise, the ethic, we won't want the tested technology will first be tested on human. And even if we lack this ethic consideration, the regulatory authority will dictate that we need to provide enough preclinical evidence to justify the technology is at least safe. And this brings me maybe to the one of the I would say reason why it's very important, even if we don't look at safety, if we want to optimize the chances that early clinical study will work, we do want, via the preclinical work that we are conducting, to get information that will enable us to predict efficacy and to better tailor the preclinical or the clinical study. This is via surrogate markers or biochemical biomarker to be gathered. What will be the first do? In my opinion, the first do is ethic, regulatory, and legally. If you have concern as to anything that had to do with your technology and potential safety, human safety, and there is no data, literature data, or previous experimental data that enable you properly to address the concern, it is a must to conduct preclinical study to address it. This is dictated by the regulator, and this is also a kind of a legal obligation. So this is the first do. With the first do coming also the first don't do. And the first don't do is don't conduct studies, even if it's cheap, even if it's free of money. Don't collect data or information or anything that you don't know what to do with the results. Sometimes gathering this type of information will just create damage because it will invalidate things later on, because it will raise question, because it can raise concern. So whenever something is being done, before it being done, the one that decide or dictate to run the preclinical study must know what he's doing with the result, whether it will be a positive result, negative result, or within the gray zone. So if we're speaking about planning, we need to plan studies or to do them in a structural manner. Structural manner means that we need to work within a kind of a framework that contains 4 proofs that we need to bring into the regulator, in order to enable him to assess the maturity and the potential safety concern of tested technology. And what are the 4? The first one is mechanism. We need to understand the mechanism, or at least to claim that we understand the mechanism and to support it. Because if we know the mechanism, we know to assess or to predict potential adverse side effect. We need to have a proof of principle, meaning that we need to justify that the mechanism that we are targeting with our tested technology is relevant to disease and we can alter it. And eventually this brings us to the proof of concept study, meaning that we are demonstrating in a model that we are altering the relevant mechanism utilizing our technology, we are getting kind of improvement in terms of clinical or any other parameters that we are evaluating. And today it's also necessary to demonstrate the relevancy. I can say that I'm not familiar with diseases that the unmet need today is complete. For any disease, even the worst one, there are some tested or approved technologies or drugs that give some kind of benefit. So when we are conducting studies aiming at demonstrating what we are going to present later on as a novel technology, we do need to demonstrate the relevancy of this technology against the existing technologies that are already in the market. All need to be done in a translational spirit. What do I mean by translational spirit? We are not going to, it's not so correct, but the aim is not to cure disease that we created in animal model. The aim is that the technology being developed will cure or bring benefit to humans. So the experimental animal data should be relevant. What are the rules that enable us to teach us that indeed the technology might have chance to work in animal as well as working in human? The first one is independency, laboratory independency. I can tell you, early stage technologies from academia, from hospitals, from institutes, from internal work done within bio and pharma startup companies, and even big one. It is quite challenging to reproduce them in a laboratory independent manner. So this is the first rule. Providing that that this barrier was overcome, and some, I won't say all, but some could be reproduced in independent manner, in unbiased manner. Then there is the rule of species independency. We do not develop technologies to treat mice. We need that it will work on mice, on rats, on I don't know, pigs, dogs, monkeys, etc. We need several species. Several species will indicate that indeed there is a good chance that it will work also on human. We need to test technology in several models. We need to, there must be understanding that disease model is not disease. Human disease model is only a very, I would say, very limited spectrum of the disease that being mimicked in an animal model. That's all. It's a spectrum. It's not a disease. It's only a slice. So in order to demonstrate efficacy, we need to be smart enough first to understand the mechanism, or to predict what is the mechanism of the technology, to be sure that this mechanism is relevant to the model that is being chosen. Otherwise, you know, it's like shooting in the dark. It won't work, but it won't work not due to the fact that technology potentially won't benefit human. It won't work since we didn't choose the right model. But should we choose model that contain these parameters, we must find another one, at least two, with a different spectrum, with a different slice of the disease. And if we match all those parameters that I spoke, laboratory independency, species independency, model independency, we are in a good position. And we always need to use relevant positive controls in each one of them in order to assess that the model work, and also as to have some kind of a ruler as to the potency of this technology that we are going to develop. Are we going to use new models, new experimental models, or old technologies, or old model or standard model to demonstrate efficacy and safety of technology? The answer is the thumb rule. The thumb rule say all technologies will be tested with new and novel methodologies, while innovative and new and exciting, I don't know if exciting is the right word, but new innovative technologies should be tested with the standard one. And don't mix between the two of them, because it's becoming a salad that you cannot really understand what is happening. Saying so, I would say that it's still art of integration, meaning that the art is that we must combine, because today if I claim at the beginning that we need to bring information as to mechanism, in studies we need to use traditional tools like histology, biochemistry, behavioral, clinical outcome, whatever, together with innovative tools like omics, proteomics, genomics, metabolomics, etc. It needs to be integrated, but it needs to be integrated in a smart manner, in a balanced manner, taking into consideration the previous point that I mentioned, not to ask questions that I don't know what to do with the result, and not to test extremely innovative things with extremely innovative tools, because the result sometimes we won't be able to interpret the result, because there are some unknown as to the technology together with unknown as to the new methodology that is being used to test it. So you must understand how to combine. You must predict the mechanism of your technology in order to choose the right model. And there is another point which I think very relevant to our Israeli audience. And as Israeli, I say, don't be greedy. You pay $1, you pay $100, you pay 1 shekel. It's okay. Don't try to squeeze too much. If you ask for too many, if you try to extract too much from a model, I want to run biochemistry, I want to run behavioral, I want to run microarray, and I want imaging 3 times if possible, and also take the liver and split it to 4 pieces. One I want immunohistochemistry, one I want fact sorting, etc., etc. So eventually you will get nothing. It's like the Schrödinger cat. You touch, you open, you look, you change. Don't mix should it possible. Sometimes it's dictated by the regulator that it is must, but if it's not must, don't mix between efficacy and safety. You're running efficacy study, focus on it. The model induction by itself is associated with things that can be considered as adverse side effect or whatsoever. And sometimes it's very hard to separate between things that are associated with the technology being tested and with things that are associated with the model, with the disease model. So whenever it's possible, don't mix between the two. And it's not generic. One shoe doesn't fit all. Each technology need to be, the preclinical package need to be planned as an innovative one. You need to know the regulation. You need to be familiar with previous or similar previous developmental programs that were approved for similar programs by the regulator. I'm using the word ping pong again, or orchestra. You need to speak and communicate with all, with the regulator, with the clinician, with the IP expert, and with the preclinical side. Only via good communication with only via balancing things, you will have a successful program. And finally, I will address the issue of the GLP. We all love GLP, good laboratory practice. I am not familiar with anybody that don't like good. But we need to understand, what does it mean? It doesn't mean good GLP, good science. No, it's not. It's not necessary. It might be also good science, but it's not, this is not the point. GLP is commitment. GLP is legal. It means certain level of degree of documentation. It means calibration of tools. It means training of the staff for certain procedures. It means experience within a certain field. It means auditing. It means many things, but it's nothing to do with science. It's a lot to do with legal. And it's very similar to marriage. I think so. it's a commitment that you should be mature to take, with the understanding that the divorce is very, very expensive. Meaning that if you decided to run a GLP study and you didn't choose the appropriate site, and there were mistakes and there were problems, the cost in terms of time and in terms of money to clean it will be high. So here the rule, pay cheap, get a monkey is a valid rule. But you need to understand also when to use, how to use. GLPs mainly for external use. It's a legal document that protects you at a later stage if in a worse scenario there are problems with any stage within the clinical phase and someone is doing reverse engineering. The GLP, if you have a good package of GLP studies, according to the directive, you spoke with the regulator, it's all approved, the GLP is the legal document that will protect you. But you need to bring into the dossier studies that are non-GLP that provide the rationale for the planning. Why you would choose, I don't know, 5 milligrams of X and give it 2 times daily. Why? Because you run 3 non-GLP studies and confirm it at a later stage with the GLP-1. So I will summarize by the statement that preclinical work is a reverse engineering. It's not a study. It's not that you are running a study and then, okay, I see the result and I decide what to do. No. It's moving from one decision gate making to another one, but with a real understanding of what is going to be the end product. The end product meaning is the stage of the clinical package that is being supported by the preclinical dossier. It's an art. It's a lot of understanding. It's not just execution. And do it properly. Thank you very much.
Thank you very much, Itschak. We'll take note. We took notes of your recommendations, and we move now to Idan Dalyot, the Business Development Manager of CATO-SMS Israel. CATO is a clinical CRO, and his lecture is going to be focused from bench to bedside, first steps in clinical trials. Please.
Yes. Hi, can you hear me, Rital?
Can hear you.
Perfect,
yeah.
Thank you. So I will start. Thank you, first of all, for inviting me, you, Joe, and the rest of the team. I think this is a nice opportunity to show some of the lessons learned and how we, as a CRO, that we provide clinical services, but by the way, both CMC and preclinical in our headquarters in the US. Those services are in our portfolio as well. But we're going to emphasize today on the clinical aspects of drug development. And just as a side note, we always talk about drug development, but basically this is much more than just drug. Nowadays, the modalities of therapies is much bigger than just drugs. We have biologics, and we have a combination of medical device and cell therapies and gene therapies. So all of those things fall when we talk about development under drug development, but there are nuances between each and every one, which we will not cover today, of course. Okay, so what are we going to talk about today? We're going to talk about translational medicine. We call the presentation moving from bench to bedside, which means basically going first in time to humans. All we've done before that is the preclinical work, the CMC, working with regulatory authorities. All of this falls under the preclinical or before human. So now we translate everything to humans' use of the drug or the therapy. We talk about the processes that govern those development aspects. What are the considerations when we conduct the first human trial? Characteristics of adaptive design clinical trial. This is an approach which is relatively new. And if I have enough time, I hope I can convey some of the highlights of that approach and the advantages. And I'm going to share some lessons learned, which is always good to hear from others what they learned throughout the years. Nowadays, there's an impressive advance in all the work and all the tools we have for preclinical work, for discovery of new molecules. Most of the companies today have a new drug, but they actually have a propriety over a platform to find out those new therapies, which may be proteins, antibodies, other new chemical entities. So there's a lot of movement forward in that respect. Controversially, for clinical trials, there haven't been so much movement or progress in the past 2 decades. There are two exceptions for that. One of them is the adaptive trial design, which I will cover today. The other one is the personalized medicine. I won't talk about this today, but it's still kind of in the beginning of that, and there's a movement towards that, but there's still a lot of work to do to get the therapy very personalized to each and every patient. It's more correct or more being done today with oncology, less for other therapeutic areas. But again, this is something that we will see more and more in the future, near future. But other than those, nothing much has changed, and we have the traditional movement of the phases of the clinical trials and the development in the clinical settings, and that's kind of a dissonance between those two areas. As a reality check, currently, development is very long. It can take up to 12 to 15 years to get the drug to market. The costs are getting higher every year. You can see the chart here from 2016. It can get up to more than 2 billion for the entire program. Sometimes it's less. It depends, again, on the indication and other aspects of the development. But it's very costly, and time to market is a very important thing, especially for the investors. And as we see and look at the past 20 years, we see less and less drugs, new drugs being approved every year by the FDA. This is somewhat discouraging, but that's the reality of it. So translational medicine, in the NIH, which is in the US, a branch of the health and human services, have a goal to improve the processes related to translational medicine and translation programs, which means, again, the first few clinical trials. This program is trying to improve the efficacy, quality, and impact of drug development processes, to develop new strategies so we can tell early on if a drug is not working, so we can make a very fast go-no decision and not invest so much time and money, which will later go down the drain. So if you can tell in advance this is a no-go, that's the best way to go and move forward with something else and not invest too much funds in that and money. We want to identify processes that will reduce costs, of course, and the duration of the development. We said that we make quicker and smarter decisions as a whole. Again, those are the goals. And there is an initiative to go around that. Now, what are the tools we have right now? We have the biomarkers that was mentioned before. Sometimes those are called, I lost it, surrogate endpoints. So you won't wait for overall survival for oncology patient to see what's happening after 10, 20 years. No one will pay for that. No one will wait that long. The money is going to be unbelievable, the cost for that. So you find other points or biomarkers that tell you and hints on a very good percentage of success and validity that you are doing something good with your drug and your therapy. So that's a biomarker. We have the translational toxicology studies and methodology that help doing those aspects. We have smart and early human flexible study design. One of them is the adaptive trial design, which again, I will talk about later on. There are new biostatistics technologies and techniques today that actually allows you to take a glimpse of your data without harming the power of the data. Otherwise, you can't do it too often because you lose the power. And then at the end of the day, the study won't give you the outcome you're looking for. So we have new methodology around biostatistics, and we have molecular genetics, again, another discipline by itself that is very useful in implementing translational medicine activities. We talked, the two speakers before me talked about the non-clinical development and the pharmaceutical development, which is the CMC. All of those together after, of course, we discovered a new drug, go hand in hand and help us to support the first in human trial. Of course, we need to have the regulatory authorities approval to do that. At the Committee, Helsinki, depends what region you're looking around. In Europe, it's the clinical trial application by the competent authorities. All of those have to be in place in order to get to the first in human trial. I won't go into length too much. I think the next speaker, Dr. Newman, is going to talk about CMC. Sorry, we talked previously about CMC, but I won't go into length. But in terms of the CMC, those things have to be established. The profile of the drug candidate, the analytical assay that you will utilize throughout the process, what is the manufacturing processes. Scalability is the utmost important thing that I cannot stress how much important it is, because manufacturing the drug just for the clinical trials for several couple of hundred of patients is the one thing, but later on, if you have a marketing approval and you want to scale up the production, it's a whole different ballgame, and you have to be prepared for that, because sometimes things fall between the cracks and you can actually move to production even if you have an approval, and that's a big no-no. You have to control the impurities and the quality of the tox in your formulation, and the pre-formulation and clinical trial supply have to be set in order to get to the clinical trial. Again, we talked a little bit about non-clinical or preclinical. It's synonym work which is needed for your clinical trial, first clinical trial. You have the IND enabling safety and tox. We talked about, do we need a GLP or not GLP? So yeah, at the beginning, we recommend not to go with GLP, because if you have to do a go, no go decision based on that, what's the point of this high cost of a GLP study if you don't need that information later on? I will go to the next item here because we don't have much time. So the estimation of the safe starting dose in human, that's the utmost important thing you have to have in place in order to allow you to go to the next phase, which is the clinical trials, which means how do you actually set the first dose, the maximum dose allowed for humans when you go to your clinical trial. And that's a big question that everybody asks themselves: what doses to start your first clinical trials? There are methodologies and guidelines how to do that. It's not a complete guessing game, but there is some thinking behind it and a way of doing it. There's a nice sentence from Paracelsus, which was a Swiss philosopher, and he said all things are poison and nothing is without poison. Only the dose makes the thing be poison. So that's a nice way to look into that. I want to share a nice story with you. This is called the Tusker case. Tusker is the name of an elephant in Oklahoma City. And researchers wanted to induce first in elephant, not first in man, a transient rage by using LSD with the elephant. And they asked themselves, what's the dose we need to put? What's the dose we need to use? They knew that for cats, 0.15 milligram per kilogram would induce that. So they said, let's be safe and let's put less. Let's use 0.1 per kilogram with the elephant and see what happens. And that was their hypothesis. What actually happened after they injected intramuscular to the elephant, he showed sign of incoordination. Eventually, he collapsed, and within an hour, he actually died after having a status epilepticus. So that's kind of a sad story, but it gives you a sense of what can go wrong. Of the aftermath, and what we realized happened there is basically that if you do the calculation, they gave him nearly 300 milligrams of LSD. And now we know that for human, 0.02 actually is enough to induce that. So the question arises, what data we need to look at, and because using that, extrapolate what doses to use. So essentially, they gave him, if we compare him to an elephant, the human to an elephant, they should have used like 6 times less. and maybe he would have survived that. Again, not a clear cut, but that was the mistake because they compared a cat and not compared a human. And now there are more sophisticated way to extrapolate the dose, the first starting dose. Of course, this will have to be backed up with the preclinical work. And if we have induced any safety concern and we want to avoid them, so we reduce the dose. But again, this has to be decided and thought of very, very carefully when we move to the first patient. One of the benefits of early regulatory interaction, and the next speaker, Dr. Mutzi, will actually talk a long thing about that, I'm sure. But there is, from our experience, some kind of a scaring feeling or some kind of antagonism to approach the FDA or other regulatory bodies if not really needed, because they're probably going to shoot us, send us back to the drawing board and change the whole thing. That's not necessarily true. We find it more and more that it is very, very beneficial to approach the regulatory authorities. And let's talk about the FDA as an example, and try to talk to them and see what's their buy-in on our development plan. And the information we get from them is priceless. It's really priceless, and it's worthwhile the effort. Even if we're not doing it, planning to do a clinical trial in the US as a first step, we still can approach them and provide information we have and advise with the FDA and get some good input. Usually this is done through a pre-IND meeting, which is nowadays not face-to-face, it's via Zoom, but on a regular days without COVID-19, you actually go to the FDA in Washington and sit down to the right people with the right discipline according to your indication and drug modality, and you get a good input from them. This is true also for Europe with the EMA, and you have the scientific advice meeting. You can have an end of phase 2 meeting, which again, another crucial step before you get to your pivotal study, which is the phase 3. And there is another way of talking to the FDA, which is the special protocol assessment. This is kind of a golden thing. If you have that, a golden egg, it's not something to get, but it basically gives you an approval from the beginning by the FDA that whatever you will do that was approved of the FDA already will eventually grant you a marketing approval relatively faster. So we move to the first-in-man clinical trial. What are the goals? So first of all, safety. I cannot stress this enough. The main goal of every first-in-man clinical trial may be a patient or a healthy volunteer will be the safety, to see what's happening with this patient, to detect any safety concern. You usually do a PK study, a pharmacokinetics, which means what does the body do to your drug or to your formulation or to a molecule. You do a single dose or a multiple dose. You need to show that you are gender equivalent, that you have 50% from each population, so you stratify your randomization or your enrollment by gender or age, and sometimes you have to show also food effects. You do the bioequivalence of different formulation. That's the right place to do that. If you haven't set up the formulation yet, you can do it in a phase 1. First of all, and you do look for efficacy signals, just a hint of some kind of efficacy and pharmacodynamics. So how is the molecule affects the body on the therapeutic level. And eventually you need to come up with a phase 1 after you know what is your recommended phase 2 dose, and that's critical. Your standard, maybe you've heard that, so that before, your standard 3+3 design for first-in-man, may it be patients or healthy volunteers, will be 3+3, which means you enroll 3 patients. If you have a DLT, which is drug-limiting toxicity, you enroll another 3. And if those 3 don't have any more drug-limiting toxicity, you can move to the next cohort with a higher dose. Eventually, you have to stop because you have more than one DLT, and then you go one step back, and that is decided to be the recommended dose for your phase 2. I'm going to show another. This is related to a different design. This is another way of moving forward with less patients, which means faster, but not put at harm the patient. You can enroll 1 patient because your first doses are really under therapeutic effect, and they are very low. You don't expect any safety concerns. So if you have nothing, even with blood patient, you can move forward. But then you say it's not just the DLT. I can have a grade 2 toxicity, which is less severe than a DLT, but it's a red flag. So this one is being used as well. And again, you can move within those cohorts, you can move again to a 3+3. It's a bit complex, but it's another accelerated way to move forward when you have many cohorts and you don't want to enroll too many patients. So we move to adaptive clinical trial design. What is adaptive clinical trial? So this is an approach that defines as a study that includes a prospectively planned opportunity for modification of one or more specific aspect of the clinical design and the hypothesis based on analysis of the data. So basically, it's not new in respect that you can do it in a very simple every clinical trial, but which means that you then do an interim analysis and you lose the power of your clinical trials, which means you actually have kind of two clinical trials. In adaptive, you can actually aggregate or put together a phase 1 and a phase 2 and look at the data without damaging or harming the power of the data. And moreover, if you do change in a regular clinical trials, if you have a change to the protocol, now you have a protocol amendment and you have to go back, do the submission, get an approval, and then enroll in so many changes, and they are very costly. On the basis of an adaptive design, you don't have to have so many amendments as long as you did the thinking in advance and you lay out in your protocol, the clinical trial protocol, you lay out all the options you might encounter, and you give yourself the flexibility to move between different ways of moving forward your product. So if you look at this scheme here, your traditional sequential trial will be a phase 1, phase 2, phase 3, but an adaptive seamless trial will be shorter, as you can see. It actually encompasses 2 phases in one trial, and you can actually have those windows, which are actually called interim look, not a lock, look, because you can look at the data in a statistical way that doesn't, again, harm the power, and you can keep using the information, the data, throughout the entire study. So that's a very big advantage. So what can be adapted in an adaptive clinical trial? What are the things that are thinking that maybe need changing throughout the life of the clinical trial and are already laid out in the protocol? It can be the sample size. I can increase or decrease that. I can use a different regimen for the dosing. I can do a randomization, which helps me, move forward with the program. If I find a dose which is has a very good outcome in terms of efficacy, I can play the winner rule and enroll more patients to those dosages specifically. And by doing so, I have a better power and a better outcome for the trial. I can change the subject criteria, inclusion, exclusion, mid-trial without a protocol amendment, again, because I thought about it in advance and it's laid out in the protocol. And you can set early stopping rules if you have no efficacy, so why invest more? You can stop and save some money. Execution of more than one trial in one single seamless protocol. Usually for adaptive clinical trial, you can do a phase 1A or 2 and 2A together, or 2B and 2A together. And by doing so, you actually cut very much on the cost. Nowadays, the regulators actually accept the adaptive trial design, but this will usually not be applicable for pivotal trials. In pivotal trials, the authorities and the FDA expect you to come ready to know exactly what will be the endpoint and how you're going to do the trial from start to end with no changes. Not to say you don't have protocol amendments in the phase 3, but usually not with an adaptive trial design. Okay, I want to share some lessons learned from us, from CATO-SMS.
Sorry, Idan, you have only 2 minutes to go.
That's perfect. That's exactly what I need. Thank you.
Okay, perfect.
So selecting the right phase 1 unit, that goes without saying. You have to know they have the experience and they have the knowledge how to do a first-in-man clinical trial, which has its own complexity. You have to select the right CRO that have done phase 1 and healthy volunteers, for example. This is critical. Data collection, you need to make a decision if it's an electronic or CRF. Usually electronic is better because it's faster in terms of cleaning the data and have it ready for, let's say, a DSMB, that's safety monitoring board, to assess the safety and give the green light to move forward. You have to have realistic timelines for enrollment and study completion. You have to have realistic timelines for points for coinciding assessment. You can't, the burden on the sites is very high. If you put too many assessments at the same time, you just cannot handle it and it hurts the data. You don't get the right clean data you want to get. Adequate upfront planning, that's the most important thing. For adaptive clinical design, you have to have a lot of planning and thinking in advance what can go wrong and now I can mitigate it in advance in the protocol, and by doing so, allow myself to keep going forward if something happened, which is maybe not wanted or not preferred, but it is what it is, but you can still move forward. And you can explore a broad range of dosages. Logistics, again, very important. For this reason, we really try and convince sponsor to do a dry run without a real patient, just to see all the systems are in place and everything is going smoothly. You have to ensure clarity regarding roles and responsibility between all stakeholders, so nothing falls between the cracks of who's doing what. Again, terminology is very important, make it fair understanding, and you have to make sure everybody understands what's going on. And again, timely generation of tables and listings from a statistics standpoint, you need those for the DSMB, and for this reason, I mentioned CRFs and electronic EDC is much better. To conclude, expediting development is not a result of cutting corners. I think all my peers here will agree with me. Cutting corners is usually not the right thing to go around things. You can find the right way to reduce costs, but it's not by cutting corners, it's by doing good science. You have to maximize learning experience with each trial, use your lessons from previous trials and put them in place when you go to the next phase. Never rush for the first patient, first visit. If you rush, you make mistakes. Your first patient will fall in the cracks. The data will not be what you expect it to be, and you will lose valuable time and money in the way. And that's it.
Thank you, Idan. I take your advice for not cutting corners. I've been doing it all my life, although it's a tedious work. Thank you very, very much.
I know it's something easier done than saying, because all the investors want to rush things, but sometimes it's going to hurt you much more if you do that way.
Talk about it also. Yeah, yes, you're right.
We know it's a challenge, but that's the truth of it.
And we move to our next presenter, Dr. Moshe Newman, the founder of Biomedical Research Design company, and he will talk about pharma medical regulation. Dr. Newman. Idan, please unshare your screen. So.
Can I share my presentation?
Yes, I need to stop sharing here in a second. Why can I see it?
Okay. Thank you very much for inviting me to give this lecture on the Pharma Toolbox webinar. I know that you had a long day today. I believe that some of the previous lecturers dealt with some of the aspects that I will show too. So I will enjoy your message if I can go faster and go to the next slide. I would like to show you what does it mean regulatory affairs during a product development in the pharmaceutical field. And I will start with a general definition of what is regulatory affairs. Actually, this is a definition that I made together from some of the American definitions and some of the European definitions. But regulatory affairs was developed from the desire of governments to protect public health. And I think that looking now at the COVID-19 period, you see it very well. By controlling the safety and efficacy of health and related products in areas of, and here comes the full list of different products which are under regulatory regulations, whether it's food, pharmaceuticals, cosmetics, medical devices, pesticides, agrochemicals, veterinary medicine, and complementary medicine. And there are those who say everything that touches your body, as well as, and this is an additional paragraph, very interesting one, because it deals with the responsibility under regulatory affairs, as well as the processes involved by companies responsible for the discovery, testing, manufacture, and marketing of these products, and make a worthwhile contribution to public health and welfare, going back to the initial objective. So this is what going under the understanding of regulatory affair, and I will try to show you what does it mean when we deal with a pharmaceutical product. The pharmaceutical development actually goes through 4 different stages. The first one is the preclinical stage. The second one is the clinical stage. The third one is actually a stage that is running through the 2 different previous research and development periods, and it's called the NDA, the New Drug Application, collecting all the documentation and evidence that you need to support your application, and actually preparing this huge document package of information that you will submit to the authorities. And from here starts the review period of the authorities, whether it's the FDA in the US or whether it's EMA in Europe. You must understand that preclinical research is a very important period during the pharmaceutical development, since this is actually the ticket that will give you the opportunity to enter the clinical phases. Here you gather all the information that you need to support, first of all, safety, and later the efficacy, and convince the ethical committee, whether it's the Helsinki committee here in Israel, whether those are the IRBs, the institutional review boards in the US, or the independent ethics committee in the EU, that you have enough material to support the safety to enter the first human clinical study. During this period, for pharmaceutical and the biotechnology product, you will have to run through the cascade of phase 1, phase 2, and phase 3. Each one of them has a different objective throughout the development of the product, and I will not go into it now. And again, all the information gathered throughout almost between 12 to 15 years for a new chemical entity and a new pharmaceutical product, all the information from the preclinical period as well as from the clinical period will build up the submission package, the new drug application to the authorities. I will try to show for a new startup that is starting to think, what do I need to know in order to plan the research and development activities and all the other requirements to support at the end the submission to the authorities. I will try to show the information that is required for you to learn and to know along the different stages to know that you can fulfill the requirements. It will start with define the product medical classification as well as the pharmaceutical class. Medical classification will go back to the list of the different products, whether it's food, whether it's fat supplement, whether it's a pharmaceutics, whether it's a drug, whether it's cosmetics. Each one of them comes with a different regulatory requirement. Staying within the pharmaceutical class will have you to look also to the pharmaceutical class, because the requirements, for example, for an antibiotics may be different from a pharmaceutical which belongs to the antiarrhythmic agents or to neurological degenerative treatments and so on. The second point is define the indication or condition for use. Actually, this is disease under study, which will determine and define the patient population that you need. It's not only knowing to say that you are going to treat a specific disease or a specific syndrome. It's the ability to define the severity of disease that will later be translated into the special requirements for the study population that will be part of the clinical study. Define the dosage form and mode of administration. Wait a minute. As for the dosage form, it defines the physical form of the dose of a chemical compound used as a drug or medication intended for administration or consumption, and the route of administration for drug delivery is dependent on the dosage form of the substance. It could be powder or solid crystal, it could be a pill, tablet, or capsule, it could be a syrup, aerosol, inhaler, liquid injection, enema, film for sublingual absorption. Each one of the different dosage form will come with a different regulatory requirement package. And for this reason, you need just as part of the definition, define both the dosage form and the mode of administration, because providing it as a topical treatment comes with a different requirement. As for providing it as an injectable or an inhaled material will be required for a totally different package of regulatory information, toxicology, CMC, and so on. Define the treatment period. What is going to be the clinical treatment period in those patients will also define actually the regulatory requirements that will be part of this period. Giving a treatment as a single treatment or treatment for a week or 2 weeks is totally different from chronic treatment. And for this reason, the treatment period in patients will require a totally different regulatory, toxicology, CMC, and so on requirements. Define the intended product market. Are you going to produce it and manufacture it and send it and sell it in the US, in the EU, in Japan, in Australia? It's true that you will see in a moment that today we are working under an international umbrella of international standards for the research and development of a pharmaceutical, but still there are some local requirements in each one of those countries and authorities, and you need to know them. Understand who is the competent authority for registration of your product. It's very easy when you come to the US, because whether your product is a food or food supplement, whether your product is a drug, a biologic, a advanced therapy, gene therapy, whatever, medical device, it's always the FDA. But it's totally different when you come with your product to the EU. The competent authority for food is totally different from the competent authority for drugs, for human drugs, as well as the competent authority for medical devices is totally different authority. So you need to understand who is the competent authority that you need to deal with. Check the existence of international guidelines, procedures required during your product development, and I will speak in a moment about international sources for those guidelines. Check for existence of specific guidelines for those specific competent authorities, whether those are specific guidelines and procedures required locally by the FDA, by EMEA, by the TGA in Australia, and so on. Look for available material monographs, method of standard conduct, and there are a lot of information that you can understand from those sources what is required for your specific product. I mean, the combination of classification your product with and a specific pharmaceutical class will let you look for specific monographs for the same type of kind of product and try to understand what you are required to do too. Look for available standards required by the preclinical stage, the GLP that I heard somebody before me talking about, product CMC and manufacturing for the product clinical grade, the GMP, the good manufacturing practice, as well as the human clinical studies under good clinical practice. All those are the international standards that need to cover the different stages of development. Look for international and local product labeling requirements. A very large chapter with regulatory requirements, starting from the primary label attached to the bottle, going up to the big storage facilities of those labeling in your warehouse of the product. Define the regulatory strategy and special pathways for expedited product development that may fit your product. And I will come to it in several minutes at the end, and I will show you, because this may be some umbrella about over all the different previous sections that I noted, and get to know the submission format, as well as what Idan spoke before me, the possibility to come for consultation with the competent authority, with the FDA, under a pre-IND meeting. The same is possible in a different mode of consultation program in the EU, but if you know all different pathways, it will help you very much to prepare your submission. Going through the different stages 1 to 4, and the last one for labeling, I recommend you to look for the FDA guideline called the Target Product Profile Strategic Development Process Tool. Also, it's a draft guideline. Also, it was withdrawn before half a year. It's still on the FDA website, and it gives you a good format of questions that you need to ask yourself and be equipped with all the information to know exactly what kind of regulatory requirements you will need to fulfill during the development of your product. So actually this guideline gives you some idea for all the different stages that I pointed out between 1 to 4, and also for the labeling of your product. The same when we speak about dosage form and we speak about technology of the different pharmaceutical products, I would like you to show there's a whole line. I show you here 2 or 3, 4 examples, but there's a whole line of specific guidelines for development of a specific product by its technology, whether it's the liposome technology, whether it's the botanical drug development, whether it's, nasal spray, inhalator solution, suspension and spray drug products, whether it's metered dose inhalator, and by the way, there's a big difference between both of them. Also, both of them are combination products, but the way the regulator looks at the container closure system of them is totally different. Knowing the competent authority, whether it's the FDA in the US, whether it's EMEA, used to be in London, not anymore, moved to Amsterdam. What is going to be now after the Brexit, I really don't know. But EMEA moved to Amsterdam. I must say that the UK was a major state, European state, that governed almost most of the legislative and regulatory requirements in Europe. So I believe that also the UK left the EU, they will still have some kind of a very great part in EMEA, like some other non-EU countries. When you speak about international guidelines, you must be familiar very good with the ICH organization, the International Conference of Harmonization, that in September 90 came about the international guidelines for pharmaceutical development. All you need is to go on their website. Everything is for free. And they are organized under 4 different categories. Quality deals with all the CMC and chemical and manufacturing of the product. Safety gives you all the understanding and requirements for the different preclinical animal studies to support safety use of the product. Efficacy gives you all the different requirements for the human clinical studies. And multidisciplinary is a new category started in 2001 and actually deals with new cross-cutting topics that have the effect on all the different 3 previous categories. You have there today 61 finalized guidelines categories. There are more than 130 different guidelines. You need to know them. You need to know which of them has to do with your kind of product, and therefore you are reliable and responsible to do it by the requirements of this specific guideline. The fact that those are, as I said, the fact that those are international guidelines means that those are the international requirements for pharmaceutical development. Either you will come to the FDA or to the EU or to the Japan or to the TGA in Australia. All of them are working under the ICH requirements. The same is right also for Israel. When it comes to specific guidelines, you need to know the specific guidelines, that some of them may be in additional to the international guidelines, whether there are specific guidelines required by the FDA, whether there are specific guidelines required in Europe. But I would like to show you some kind of a trick. If you would like to look for a specific indication that you develop your product, especially in the FDA website, look in Google, look in Google under FDA guidance for industry, developing products for treatment or developing drugs for treatment, and put in the indication that you are looking for. There are hundreds of guidelines of this type. The specificity, I mean, the very unique kind of this kind of guideline is that it gives you all the requirements through all the different stages of the development. If there are specific preclinical requirements, they will appear here. If there are specific CMC requirements, they will appear here. And mainly, they will give you the synopsis for your clinical protocol. They will define the endpoints. They will give you a lot of information for the statistic sample size that you need. So this is a basic tool that you need to know when you go into a specific development under a specific indication. Mainly for CMC.
Sorry, Moshe, you have only 2 minutes to go.
Okay, I will run through the last slides. For the different CMC and method standards, pharmacopoeia, whether it's the USP pharmacopoeia, whether it's the United States pharmacopoeia, whether it's European pharmacopoeia, British pharmacopoeia, all of them are known as international pharmacopoeia, and they will give you all the data that gives you the possibility to understand, identify, and characterize your materials. Working under the international standards, whether it's good laboratory practice, safety studies in animals, whether it's the good manufacturing practice required for the batch that will serve the clinical stage. And here comes the rule. If you fulfill the requirement of the triangle basis, you will be allowed to submit to the Helsinki Committee the protocol for the human study that needs to be done under good clinical practice. Different expedited programs. The FDA have a full list of different expedited programs. You need to know them. You need to understand whether they fit your product. Go to those two different guidelines. Those are the last two guidelines that were published in this area, and they deal with all the expedited program for serious condition, drugs and biologics. And here you have the additional part published in 2019 for all the advanced therapies, gene therapy and cellular therapy. Submission. We are working now under the Common Technical Documents Organization, which is actually based from the raw data, whether it's CMC in the quality, safety in the preclinical and clinical data, the summary layer, which is the main layer for decision, and Module 1, you see, is regional administrative information. Those are special requirements for a specific area. Again, the two main guidelines for organization of all this package. This is the basic, but there are another 5 different guidelines for each one of the different requirements of the submission. Thank you very much.
Thank you very much, Dr. Thank you very much, Dr. Neumann, for very, very helpful and useful information. And we quickly move to the next speaker, Dr. Zaki Nudelman, Business and Development Advisor from Israel, will talk about guidelines for obtaining funding. Please, Dr. Nudelman, the floor is yours.
Thank you, Revital. Hello, everyone. Okay. So can you see my screen?
Yes. Yes.
Okay, so I will be talking about guidelines for obtaining funding for early stage companies in the field of pharma and biotech. Shortly about myself, I was born in a beautiful city, used to be called Leningrad, St. Petersburg today. In the 90s, moved to Israel. I'm a pharmacist. During my academic phase, I was developing drug delivery systems for transdermal drug delivery, controlled release drug delivery. And I'm also holding an MBA in biomedical management. And I work with numerous companies in the field of pharma, of natural products, cannabis, and also TTOs, technology transfer offices in Israel. So I would like to start with a very nice episode from Alice in the Wonderworld. When Alice meets the Cheshire Cat and asks him which way should I go, and the Cheshire Cat says that depends on where are you going, and Alice says I don't know where I'm going. So the Cheshire Cat says then it doesn't matter which way you go. So when we look on this dialogue and try to understand how we apply it to pharma and fundraising, so we should understand that we do need to know where we go, and then thinking what is the best path to choose and go to the success of our venture. So I guess that people who join this webinar do know where they go. So I will try to be your Cheshire Cat for the next 15 minutes and share my experience and recommendation on how to approach the fundraising process of early stage venture. So let's talk about the biotech, not Alice Wonderworld, but biotech Wonderland, sorry. And who are the characters here? Who is Alice in our world? And let's look at these groups of characters. So first we have the academics, then we have the entrepreneurs, we have investors, we have governments, and we have the large companies, the big pharma. And we need the next thing that we need to do is understand what are the goals of each group. And when we talk about academics, so it's basically to make research, publish, get grants, contribute to science, and that's what is happening in academics. And it's also important to understand that all academics today have the TTOs who commercialize the science that is developed in universities, and it is very relevant for early stage startups who many of them which starts from the academia. So the next group are the entrepreneurs, and so what are the goals of entrepreneurs is monetizing the idea, raising funds, developing the product, and creating value, and also publish what IP, more relevant IP, because it's something that is critical for pharma venture. Investors are the most simple. They just want to get high return on investment, so-called ROI. Okay, governments, they encourage the economy, maintain and improve the health of citizens, and sometimes it's theoretically, but I'm going to show that it's also practical, not usually theoretical. And the large companies who deliver the products, increase their market share and generate the revenues. So this is the final stage of product development when we get to the market. So who are we in this ecosystem? So we are entrepreneurs who want to develop our technology, our idea. And again, I'm not excluding the academics from this list. And who are our clients? Who are our actual clients? Are investors, governments, and large companies. These are the organizations and bodies that can finance and advance our project. Okay, so let's put the pieces together and talk about what should we practically do, how we start to run our idea, to run our startup. After we define what is the invention and what are the goals, we need to understand how we communicate our idea, our business to the investors and the interested bodies, and what are the formats that are available for us to communicate the idea. So there is a lot of formats, and we need to understand each of them. So the first one is 1-pager or 2-pager. It is a short graphical document that reflects the most essential part of your business, and the point is to raise interest when you send your 1-pager. Then we have the investor pitch deck, which is a presentation that describes your business, your venture for all aspects, and I will elaborate on this in the next slide. An executive summary, which is also a short written document that summarizes your business, and it kind of investor pitch, but in written and very short. Then you have a business model canvas or business model. So each business should be very clear about the idea how the business makes money. Okay, so finally we need to explain it, and what are the models, how we are going to develop, to commercialize our product, who is going to use the product, who are the influencers and who are the buyers of the products. Okay, then we have other formats like video, like animation to describe what happens when we inject something or influence some cascade, biochemical cascade. So it depends on the technology. A business plan. So a business plan is a very elaborate document that describes your business and all the aspects. And an important thing about business plan is that if you try to compose your business plan at the very beginning of your venture, it will be very inaccurate, okay? And it will not have much value at the beginning, okay? You need to be very clear about your ideas, your decisions, and everything that you do in your venture is a decision, and each decision has consequences. So after you are clear, you need to write it down in a very accessible way, and this can be used with advanced negotiation with investors and other bodies. Conversations. So during the Corona, unfortunately, we don't have much opportunity to meet people, but yet we have the opportunity to Zoom and to have video calls, and it is important to talk with people, to talk with investors, to talk with even other colleagues that are not investors, and it's all about network and meeting people and sharing your idea with other people. Another interesting format is an elevator pitch. It means that you have a very short time to deliver your idea, and you need to choose very carefully what are the messages that you are delivering in a very short time, okay? So anything that enables you to communicate your idea, to tell about your project, your technology, your plans, it's the way you should act. So about the investor pitch deck, which is I think the most known way to communicate with investors. So if you just Google, you will see a lot of templates that most of them are similar and in this way or another. And you can be very creative. Despite this, you can be very creative with your investor pitch and have the information flow in a very accessible way to deliver your idea. And the fundamentals of such pitch is covering all the aspects of your company. So, for example, introduction and your team. So you should stress your expertise, your previous achievement, because the bottom line is that when you talk with the investor, it's a personal interaction, okay? And the investor should understand that he deals with a good team, good people, with achievements, with motivation. So this is the beginning. Okay, then we define the problem, or called unmet need, or any other definition. So you describe what is the problem, what would you like to solve, okay? And then you can explain your technology, how it works, how it better than other technologies, how it needed, okay, why it's needed, and also describe your product, how you see the product. And we've seen that in previous lecture of Leonid, he talked about the delivery system. And it is not enough to have, I totally agree that it's not enough to have a good molecule that works. You need to deliver it in a proper way, in a quality way, and you should define how the product will look like. Is it a tablet? Is it an injectable? Is it a cream or dermatological product? And then you should talk about the market. You should know the market you are acting in. Also talking about the competition, knowing the competition, knowing the current solution for the problem that you are targeting. And regarding the market, it is important to understand that there is a market value and there is a market potential. And the intuitive way is to say, I would go for the largest indication. There is a lot of money in this field, and this is the best choice, but it's not always the true. You should look at the potential where your solution or your technology will have the most advantage comparing to existing technologies. And even if the indication is smaller, then it might be more suitable to choose this indication. And many companies today even go for orphan indications because it is easier to show your advantage. The regulation is also easier for orphan indications. So it's a good go-to-market strategy to start with orphan, for example. Business model, as mentioned before, you need to be very clear about how this business will be profitable, and also talk about what is required for the company. So after telling all the aspects and the story of the venture and your plans and decisions, you should define what do you actually need and when you need, in order to develop the idea, develop the venture. Okay, so this is a kind of very general, but each of the paragraphs requires a lot of thinking and understanding, also of technology and also of strategy of development. Okay, so it's right for many industries, but what is so special about the pharma or biotech industry compared to others? So first of all, it's a very technological, highly technological, IP-driven industry. So in order to start something, you need a strong technology and you need a strong IP, because if you don't have IP, most probably that you have no go. Okay, so this is the first thing that is critical about pharma startups. Then we talk about the barriers. The barriers also very high in pharma. As mentioned, regulation is very tough, long, expensive, preclinical, clinical phases, CMC. Then we should understand what we do with the reimbursement. Who will be paying for this drug product? Okay, is it the patient? Is it the insurance company? Is it the government? So you need to understand this and be very clear with this message. And then we talk about the market access. Let's imagine that we have all the money in the world and we can develop the idea, but we need to deliver the product to the patient, and the market is ruled by big pharma companies, and you need to access this. Okay, so it might be a licensing of your idea or an exit, but most probably you are not going with the one who sells the product at the end of the way, and this should be understood. Okay, this business is very expensive. Even for proof of concept stage, you need to perform a lot of experiments. You need special equipment. You need the right people to perform it. So it's also kind of a barrier for pharma company. And for this specific point, it is important that the other way to achieve the POC is to cooperate with academia, which already has the POCs, okay? And see how this cooperation can yield something valuable to start the journey. Also, an important thing is a due diligence. So when you talk with the investor, with the company, and it is positive, and finally you need to bypass the due diligence process, which is done by experts from the investor side, and this process is very challenging because you need to build your data that you have, to organize it in a very specific way that it will be accessible, that it will be in good order, that it will be solid enough in order to pass the due diligence successfully. Okay, so it's mainly technology, and once you are in a due diligence process with a pharma company, for example, it means that the market is not a question anymore. Okay, because the pharma company knows much more than you about the market, and if you are there, it's a very good sign, and you need to focus on the technology and explain.
Nodelman, I'm sorry, but we are running a little bit
okay.
late. So if you can kindly conclude in 1 minute.
Yeah. Okay. So we
okay, thank you.
High risk versus high return industry, and shortly about the options that we have to raise the money. So we talk about grants and public funding, like governments and non-profit organizations, angels, incubators and accelerators, family offices, foundations, venture capitals, which are financial, or corporate VC, which are strategic investors, and the key industry players. So for early stage, I would recommend to approach the first part of the list, okay, which is more suitable for early stage companies. And for example, an Israeli Innovation Authority is a good place to start with a very early stage. You can get about 1,000 shekels for each year. Then you have the incubators program, which is 8 million for a few years, which is much more significant to achieve, to create more value. And I would like to finish with tips. So do network, do your homework, know your investor, check about the previous investments and the connections. Be focused on your idea. So you need to define what is the indication, and as mentioned before, it is very important to understand where is the potential, okay? Highlight your technology, and you need to have a good development plan, and this is how you explain and mitigate the risk of the project. And final recommendation is determination. It's a very, very difficult process to run a venture, and you will have a lot of problems. You will hear a lot of no's, and you need to be determined and stick to your way and believe in it, and then you will be successful. Okay, so thank you for your attention. Thank you very much.
Thank you very much, Dr. Nodelman, for this very interesting and important talk. And now we are moving forward to our next speaker, who will be speaking about licensing in pharma, Dr. Eyal Bressler, from the CEO of Dr. Eyal Bressler & Co. Please, Eyal.
Are we okay with the slide?
Yeah, we can see the slides and we can hear you.
Perfect. Thank you very much, Gilad and Revital, for this kind moderation and the webinar, and thank you all the attendees and the presenters before me. Thank you, Dr. Nodelman, for presenting the very interesting and very elaborating investment dashboard you present, which is very interesting. I'll try to make this story short. let's begin with the Israeli law, which is, let's remember the Israeli law, not the American law, not the European law, the Israeli law, that we have two kinds of laws running in the atmosphere. One is the real property law, which is the Nadlan in Hebrew, or the land law. And the other law is a personal property, or metaltalin in Hebrew, law. There are a lot of differences between those two laws. The real property law said that you cannot have ownership on land if the agreement is not by writing and it's not written in a book. Nevertheless, in personal property, there is no need for written agreement, and there's no book. For such, the intellectual property is divided between two of those groups, where utility patents, as we know as patents, design patents, as we know as designs, trademarks, and patent breeders' rights, are much more in the Israeli law like McCarthy, like the property law. So you have no rights without written rights as an agreement and written in a book, where the personal property law governs the copyright in Israel, but not in other countries like in the state, and the reputation, trade secret, etc. Some of the laws and some of the rights you can find in the world, like utility patents and trade dress, are half and a half. So utility patent, which is utility models. Sorry for the mistakes. Sorry for the mistake. Utility models, which are really a big issue in Europe, in China, and Japan. Let's say that 50% of the filing of patents in Japan and in Germany is utility models. Half and half. They are quasi patents, so there are no really registration of the assignment, only in the book, but only in some of the countries, and so forth, by trade dress, which are kind of elaboration of copyrights and trademarks, which are half non-registered and half registered, so they are quite difficult laws. So let's talk about an example of a toolbox. Let's talk our beloved Zifandil citrate, which is known as Viagra by Pfizer. So let's say that we want to have the strongest and the prolonged protection of this technology. So we can file patents. The patent was filed in the middle of the 90s and was accepted in the 1906. And the patents were available for this molecule and for the treatment for erectile dysfunction in males up to the last 10 years. And then the patents are dead because the patents are valid only 20 years. So patents are very expensive, yet very, very strong protection, which dies after only 20 years. So we have the design patent. Design patents are valid for 15 years in the state and 25 years all over the world, except the state, also in Israel. And the design patent is very important to Viagra because everybody who uses Viagra knows that the diamond shape of the pill, so you can protect the diamond shape of the pill as a design, you can protect the 3D trademark of the pill and the Viagra blue color shade as a trademark, the Viagra wordmark, etc. And those trademarks last forever. If you're not killed, then they will stay forever. So when you say toolbox in the world of licensing, the toolbox is of the properties of what you want to license. The properties can be patent related properties, design related properties, or reputation or trademark related properties. So what about those properties and how are they distinguished by year? So this is the number of patents people are filing in the world in Viagra in the last, let's say, 10, 20 years. And you'll see that every second day, a guy is filing a patent in Viagra in the world. So there are a lot of patents in Viagra, even though the poor and miserable Pfizer which gains only $5 billion a year as a revenues for Viagra, they lost the patents. They didn't file patents in the last 20 years after the attempt in 2008. So there is a first wave of erectile dysfunction that protected by patent as a differential threat or as a method. But who are all the others people that filing a lot of patents? On the left, we see that Pfizer, as we said, filed the patents, 25 patents in the hot year of 2003, and then disappeared. And then newcomers are coming, like emetics, biotics, etc. Those are coming for new aspects of the Viagra, new preparations, new uses, like use for pulmonary hypertension or treating for the Raynaud phenomenon, etc. Second use, as we call it in patents, for Viagra or second methods of producing the same. And a very interesting, as you call it in Yiddish and in Hebrew, a planter, we'll see in the last few years. In the beginning, the number of assignees and number of patents were much similar. So we have a assignee for every patent. And this was quite linear up to the recent years. Then in few years, and 10 years ago, there are much more assignees than patents, and nowadays there are much more patents than assignee, what we call coalitions of license. This is what the term in economics, coalition of the rights, and we are here. So there are less and less assignees and more patents per assignees. And who are the newcomers? Tony Weinschneck is one of the comers. He had filed about 30 patents last year about the new use of Viagra. So we can see that what we are teaching in our webinars it's much working. Nobody knows nothing about the fertility threat in the years up to 2003. This is the lag, where no patents, no technology, no money, no credit in this technology. And then one guy, Peter, we'll see him later. Oh, we see him here. This is Tony, and Peter, the guy who invented the fertility tract, disappeared after a few patents. He's not relevant today. So in the log, one genius guy understands what to do with the molecule, and the production, the selling, the number of assignments and number of licenses in this technology is rising. And then there is a plateau. And in the plateau, some technologies are still running, and some plateau are going to be dead. So the average is a plateau. And in the plateau, there is another way of another status quo, where in the log, the status quo is follow me type business plans and follow me type license. And we'll talk about it later. And in the plateau, there are me too types of business plan, meaning I can improve the product. I can reduce his production price. I can make him safer, etc. And then there is decline of the first generation. And when the first generation dies, the second generation arises, and then the third generation. So the accumulated amounts of the fertility threat in the last 20 or 30 years is like so. This is the sigma curve, meaning that the fertility threat is evergreening itself. So about the licensing, you can see there are peaks in licensing. There's the first generation licensing where Pfizer never licensed the product. It was Pfizer product. And then the second generation, now we're in the third generation. And this is litigation. If you cannot license, you litigate. So there are the peaks of the litigation, and there are more and more litigation those days than before, because now we are on decline of the first and second waves. So what are license? License, and in the right hand you see the Israeli law in Hebrew, and the left end you see the Israeli law in English. So everybody can read what they prefer. There are two types of license. The first is exclusive license, and the second is non-exclusive license. Non-exclusive license, you permit the commercialization of a technology under a patent for one or more stakeholders or one or more players in the market. Whereas in exclusive license, you deliver your rights to another player and is on the only one who can avoid other for commercializing a technology. I'll say it in other words. Patent is a right for its owner to avoid or eliminate or sue or kill other players in the territory for 20 years in a certain technology. Now, patent is a license to kill. When you have a non-exclusive license, it's really a license that I will not kill you. This is a waiver, I will not kill you. You can commercialize, I will not kill you. Please give me between 2 to 6% of your revenues because I'm not killing you. But this is just a waiver, I will not killing you. Whereas an exclusive license, the guy who got the exclusive license is taking is under my shoes, and he is now the relevant player in the market. I want to warn you about joint ownerships. If there is one say that you should remember, avoid joint ownership. I will explain why. When you have joint ownership on a patent, each party may practice the commercialization of the technology in the country and import from other countries without prior consent of the other player. So let's say that a strong guy, let's say a Goliath, and a weak guy, David, are joining together as a co-owners in a patent. So Goliath can do whatever he wishes in the market without prior consent of David, because he just can't do it. Now, if you want to argue, says there is a contract between us that the contract avoids you, as the paragraph says, we're going to a court, then Goliath usually kills David in the court. So joint ownership is not the correct solution for gaining money from your patent. Licensing is much better. Now, if you see the guy, this guy says, this is the way I'm going to my wedding. This is a commercial. You see, they've got suspenders and they've got a belt. Now, agreement and ownership are like suspenders and belt. So licensing is an agreement, and an agreement is a weak proprietary, is a weak means to hold a property, where a property, kinyan, like ownership, is is much stronger than a patent, than agreement. Sorry for that. So something about administrative issues that we are in the Bressler group are very, very active. You can register in the book license that everybody will know that a license was given. So if David is a licensor and Goliath is a licensee, you can write the name of both David and Goliath in the book of the patent, and then everybody will know what is the situation, and then we can register an agreement in a property manner, not a proper manner, in a property manner. Now, my best part of the show is my last slide. Not because it's my last slide, because just because I'm to deliver you to Dr. Joe Wyse, was speaking just after me. Dr. Joe Wyse and his team, I must gossip about him, have licensed, sell, and bought IP. I'm talking about patents in a value which is exceeding $50 billion at the last 5 years. So Joe, Gilad, Revital, and the group knows much about those worlds of license. I want to tell you something about my opinion about buzzwords that are known in the art, about copying, know-how, etc. So on the left, those are the buzzwords. On the right, this is what I think you should know about them, because sometimes I think terms are changing the meaning at time. So copying in Israel is allowed. You're allowed to copy, unless this copying is breaking the law. So usually copying is allowed. Use of know-how. Use of know-how is no right to sue you. If you use somebody else's know-how, is not in Israel a lawful item to sue you, unless you unlawfully breaking a contract or a law. A patent is only a license to kill for 20 years in territory. Now, about confidentiality. Know-how which is not in the public domain is confidential unless you not proactively and continuously kept it as a secret. So people says, if I write on a paper, it's confidential, it's confidential. This is not true. Confidential matter is a know-how which is not in the public domain, and you proactively kept it as a secret. MNDA is a mutual non-disclosure agreement, and this is a very rightful way to settle both know-hows of David and Goliath. NDA is a tool for the strong guy to be stronger and the weak to be weaker. So in this manner, strong and commercial entities never signs NDA. So there are no NDA and the reverse NDA. No NDA if you want to get money or to want to have a joint venture with a big company, it says, I will not ever sign you an NDA. Or there are so strong companies says, I will sign reverse NDA. What is a reverse NDA? Reverse NDA says that you sign me, that everything you tell me is not a secret. Or if it's a secret, you file the patent on that. Now, in most cases, when we're strong, we file an NDA with a non-competition clause within the NDA. And some of our clients sign NDAs without attention that they are signing a clause that they will not compete the other side. Waiver and NCA. In many cases, the waiver and ACA says, in waiver, I will not sue you, and non-competition agreement, I will not compete you. MOU. MOU is an agreement, a valid agreement. It might be able to sue you. In many cases, our clients says, I will sign an MOU with another party. It's just an MOU, just a memorandum of understanding, just what we think to do. No, MOU is an agreement. An agreement is this beginning of the end of the smaller company. This is the strongest point in my lecture. I want you to remember about the joint venture. The joint venture, the strong is usually allowed to kill the weak in a certain scheme. The scheme is the two companies sends to inventors. So there are two co-inventors. If there are two co-inventors, there are two co-signees. If there are two co-signees, there are two co-owners and bank. Because in joint ownership, each of the party practice the commercialization of the technology without prior consent of the other party. So joint venture is just a kill of the weak side. No exclusive license, he says, you pay me not to sue you. Sorry, non-exclusive license says, you pay me not to sue you, the first guy, the second guy, or the third guy, because there are many sides for non-exclusive license. An exclusive license, you pay me not to sue you, you pay me, but your stance in my shoes and you are replacing me. Assignment and sale, you become me, now you are belonging to me. This is my last slide. I feel that in my 25 years of practicing the law of IP, people mix between all those measures. You cannot understand how to make a good license agreement without understand that people can copy you, can use your know-how. If they can use everything which is not patented by you, if they can use everything which is not confidential, namely if you're not kept it confidential, and they will not license you if you sign them a non-disclosure agreement with a non-competition clause. So you must be very wise in the beginning to spread the butter on the bread afterwards. Oh, thank you very much. I really want your questions and your assertions, and my email and my cell phone is here to you, and just call us whenever you need us, and we'll be here. Joe, I think this is your floor now.
Hello.
Yeah, Joe, we can hear you now. Excellent. Okay.
Great. Okay. Let's just. Well, thank you, Eyal, for that very, very important discussion, that very important lecture. I'd like to actually, I should have called this some patent highlights. I just want to make some observations here. According to the WHO, the most prevalent causes of death on the left, cardiac artery disease, stroke, lower respiratory infection, COPD, and the rest of it you can all read, cause millions and millions of deaths a year. So we want to ask ourselves the question, for example, how many patent filings are there in these areas, and what can we learn from them? So if we take coronary artery disease, we can see that about 20 years ago there were a lot of filings. Now there is some decline, but there's still a lot of interest in this area, and we all know, unfortunately, just how prevalent this disease is, and so there are constantly new innovations and new inventions. In this particular subject area, there are fewer assignees in 2020 than there were in the 2000s, probably indicating a coalescence of interest. However, if you take the top 10 patent owners, the field is not really dominated by a few companies. If you take all the top 10 together, there's an assignee concentration of only about 35%. So that's an interesting indication for young startups. There's plenty of room. New companies who've never filed patents before have only started filing in the last 5 years. We had a quick look. There are some, You'll see in other subject matters there are a lot more. If we want to know what is being done and who is doing it as far as coronary artery disease is concerned, we can see this in this slide here. Now, here's something nice. When we understand that there's a lot of in-licensing and out-licensing going on, and there really is in this field, you can see, you know that there's transaction action. I call it transaction action. It means there's money. And if there's not transaction action, as Eyal said, there's litigation aggravation. That also means money. So there's plenty of action, money action in this area. So that should be very, very encouraging for young startups. And you can also see that the litigation is increasing, recently. It's always good. You know, lawyers and patent attorneys love litigation, but it's also a very good signal to young startups that there's money, there's value, there's interest, ferocious interest sometimes. Now, I've taken together all the I just wanted to just run through that because I know it's towards the end of the day. I just want to show you before I go on to the next part of the talk. I've concentrated together in this one slide, the top killers. On the left-hand side, I just showed you the coronary artery disease as a sort of a model. If anyone wants more details, they can of course talk to me. But the take-home message here is that we are living in glorious times if we're a young pharma startup. And why is that? It's because you can see that there are thousands of patents being filed in almost all of the fields of the top 10 killers. There was a golden era in 2010, as you can see. There were more patents filed then, but nevertheless there are a lot of patents. The assignee concentration. If you look at this column here, the assignee concentration. The assignee concentration here is not big, and in fact, again, if you take something like Alzheimer's, where a few years ago 90% of the patents were owned by the top 10 companies, now that's dropped to less than half. Again, these are all indication. These high-level datum are all indications of that should be very encouraging to young pharma startups. New patentees. You take a field like, again, let's go back to Alzheimer's or diarrhea. A lot of people forget about diarrhea. There's a lot of diarrhea around. There's a lot of tuberculosis. There's a lot of cirrhosis. Lots of patents and quite a few new patentees. Licensing agreements that we've been able to pick up. There's quite a lot of that and quite a lot of litigation. And I bet you there's a heck of a lot more under the radar that I haven't been able to find yet. So let's leave that now. Because it is coming to the end of the day, but I do want to talk about another area which is personalized medicine for patients. Okay, we all know, those of us who've slogged our way through War and Peace by Leo Tolstoy, that he invented the whole idea of the whole concept of personalized medicine. Scientists and computer analysts and informatics geniuses and molecular biologists notwithstanding, because Leo Tolstoy created the prior art well over, something like 150 years ago. He said, no disease suffered by a live man can be known, for every living person has their own peculiarities, has their own personal, novel, complicated diseases that are unknown to medicine, a disease consisting of one of the innumerable combinations of the maladies of those organs. And that is all about personalized medicine, AI, big data, and omics. So I'm looking at patents. This is very interesting. If you look at big data in the medical field, patient data, pharma data, medical data, that kind of data, the applications are increasing, but the grants are going down. Why are the grants going down? Because it's getting harder to say something novel and inventive. Yet, we do know that this field has not fully borne fruit yet. It's not fully it's not fully being exploited. So the technology, so the world is crying out for new technology. That's what this graph says to me. We look at the area of omics, okay? More or less the same story. Omics is a little bit newer, that whole idea of genomics, proteomics, transcriptomics, microbiomics, and any other omics that you can think of. This is a newer area. Grants are difficult to get. Data is difficult to get hold of. And that, of course, was in the previous slide. The two things work together. We've got you know, some sort of coalescence going on in both fields. The top assignees in they're not exactly the same players. The top assignees in AI and big data for pharma, you can see that list here. The top assignees for in the world of omics is a different you know, a different team of actors. I wonder if, you know, how and whether they'll actually get together. But this is a beautiful slide. The assignee concentration is dropping precipitously. That means there are superb opportunities for startups in big data. Same thing in omics. New entrants, boy, they're moving in. Lots and lots and lots of new companies are moving in, filing patents, companies that weren't even heard of 5 years ago. We're talking about a lot of material moving through, a lot of patents, a lot of applications moving through. Same thing with omics as well. What are they doing? What are they doing in data? They're putting together image data, patient data, devices, treatment plans. All of this stuff is going together and promises us wonderful new therapeutics for the future. In omics, we've got a lot talking about the end product. The drug will be a specific personal drug for the specific personal individual. That's what this is all about in the world of omics. Out-license already, even though there's a paucity of granted patents, there's plenty of in-licensing and out-licensing action going on. And this reflects almost everything in our practice. Almost all of Eyal mentioned the kind of transactions that we've been a part of. Almost everything that any of our clients have done has usually been long before the grant of the patent, long before, usually at the basis of patent applications, patent portfolios. Okay, same thing in omics, loads and loads of licensing issues. There's even some litigation. They've already started fighting. There are quite a few punch-ups going on in patent data, pharma data, and of course also in omics. Again, this is just another representation, slightly more granular, of what's being done in this area. Same again here. So basically, I've come to the end because it's terribly late, and I know that whoever survived this far in this sort of Darwinian Darwinian selection that we've imposed upon you, I would like to just say it's been a thoroughly enjoyable afternoon. It's been fascinating. We are all very, very grateful for these superb speakers. And it's not just a question of the fact that the speakers were superb. These are all leaders in their field. These are all leaders who any small startup that's beginning their journey must be in contact with, because these are the resources that you can draw upon in order to move forward, in order to get your invention one day at the bedside of a patient and a business, into a business. So on behalf of Eyal Bressler, my colleague Revital, Gilad, I'm very grateful to all of the speakers and to all of you attendees. You've made this all the effort worthwhile. We look forward to receiving your questions, your requests, your criticisms, your remarks, your demands. And COVID or not, we will be presenting more webinars in the future. We do look forward to meeting you virtually and hopefully soon in person. May your research-driven startups bloom into fine businesses and flourish in 2021. We are all, as always, at your service. And that's my word from Team Bressler and my promise to you. Okay. So.
Thank you very much, everyone. Good night. Bye.
Thank you. Bye-bye.
Bye.